Tuna 4. Possibly a low base.
Welcome everyone. You are viewing live video from remotely operated vehicle SuBastian deployed off of research vessel Falkor of the Schmidt Ocean Institute. We are currently diving on Malay Reef of the Great Barrier Reef about 400 kilometers from Cairns, Queensland, Australia. I'd like to introduce Dr. Brooke of Geoscience Australia and Dhugal Lindsay joining us remotely from Japan via Zoom. And Brendan, take it away.
Yeah, thanks very much, John, and welcome everyone as we dive the depths of the Coral Sea Marine Park. The seventh dive of the Seamounts, Canyons and Reefs research cruise on Falkor. Today, we've got two key components of the dive. The first is the midwater component, where we're descending relatively slowly for the ROV, observe and also hopefully sample some species of interest, plankton and jellyfish component of the dive. We hope to reach a depth of around 630 meters, and once we're on the seabed, have a bit of a look around and potentially sample some corals and other types of deep water species that we're interested in on this cruise. Once we're all settled on the bottom, we'll start the second part of the dive, which is ascending up the very steep northern margin of Malay Reef. As we ascend, we're acquiring very high resolution video and stills images, seabed geological features, and also the benthic communities, in particular certain species of hard and soft corals that are the focus of this survey. Very happy to now pass over to Dr. Dhugal Lindsay. Dhugal was unable to join the vessel because of travel restrictions related to COVID-19, but we're very happy to have him join Falkor live via Falkor satellite communication system. And Dhugal is our expert in plankton and jellyfish. So over to you, Dhugal, in Japan.
Thank you, Brendan. Oh, here we have on our screen, just as we come in here, it looks like a siphonophore, perhaps. This is one of the gelatinous organisms that occur in very large numbers in the deep sea. When there's not much food around, things are better if you're made of jelly and you don't need that much organic matter to become large. Oh, wow. It looks like a physonic siphonophore with the nectosome contracted. Video this for a little while to try and get a positive ID. It looks like there's a ball of semi-transparent swimming bells or nectophores surrounding the animal. And that area inside, I need to see if that is a float, a pneumatophore, or something else. Two possibilities occur to me now. It could be Vogtia or Hippopodius, one of the calicophorans without a swimming bell, and that internal mass is actually a parasitic amphipod or something like that. Or that could be the float, and it's contracted itself more than I think I've ever seen a physonec contract itself. See if we can tell from the video there. These siphonophores are basically colonies of polyps. Polyps for normal jellyfish look like tiny little sea anemones. But for the hydrozoans, which is the group that the siphonophores belongs to, they can be all sorts of different shapes. You have polyps that are specialized for feeding, polyps that are specialized for defense, polyps that are specialized for reproduction, etc., etc. And in a siphonophore, the polyps have evolved so as rather than attaching themselves to a rock or wood or mud, in order to live in the deep sea where there's no substrate to attach to, they attach to themselves and they become a self-sufficient floating or swimming colony, with each of the polyps having a different function. So you have some polyps, their function is to swim, others to reproduce, others to digest, others to taste. And they're all swimming around together, each with their own job. Fondly, these animals can reach the longest lengths of any animal on the planet. The longest siphonophore reaches a length of 40 meters, is the maximum that's been recorded. That's twice the length of an adult blue whale. And the word plankton does not refer to a taxonomic group. It refers to anything that cannot swim faster than the currents surrounding it. So if it's floating and cannot swim any faster than the currents, it is plankton. So a 40-meter long jellyfish is also plankton. I guess if you cut the tails and fins off a whale, it'll be plankton as well. But you get my point. It's not a taxonomic group at all.
Yeah, great. Thanks for clarifying that, Dhugal. That was really useful.
So we need to get...
Yeah, we should get going, Dhugal. Is that okay?
Okay. Yeah, that's okay. The video here is being recorded in 4K high quality codec. So I'll be able to look at that animal back in the laboratory image analysis lab and go through frame by frame to find the characters I need to identify it. Right now, because we're zooming in real time and I'm trying to identify these animals using slightly jumpy video here in the mountains of Japan, I may not be able to ID all the animals, but hopefully with this high quality video that's being recorded on the ship, I'll be able to look at them all later and tell what they are. We've got a variety of different cameras here on the ROV SuBastian. The main camera that we're looking at now is a 4K video camera. So that's 4 times the screen area of a normal high definition television camera. This will allow us to look at the very fine detail in the morphology or the shapes of the animals that we're looking at. We also have a high resolution stereo camera set attached to the SuBastian. And this will allow us, by mapping pixels in the left camera and the right camera, to say pixel number 1030 is the same as pixel number 1063 in the right camera. And we can map out the 3-dimensional space and in this way tell the sizes of organisms and the numbers of organisms within that volume. So this gives us quantitative data to go with the positive IDs we'll be collecting with the 4K video camera. Just before we started streaming, we had something called a calibration board, which is a white and black checkerboard, which we were waving around in front of the cameras using the robot arm of the manipulator to make sure we have good calibrated data for this quantitative work. Oh, we have a jellyfish in the center screen there. Let's see what it is. It looks to not be a jellyfish. This is a pelagic tunicate, a larvation, which that white spot in the middle is the body of the animal. It should have a transparent tadpole-like tail attached to that body. I think that's what I'm seeing. The image is kind of dark. And the filter feed, the filter around it is a large mucoidal filter where it filters the seawater to extract the, oh no, it's not, it's a, oh wow, that's a very transparent cell, which is also a tunicate, so it has a lot of similar body structures, but it looks to be a very transparent cell. Wow, what's it? And that mass may be the buds of starting to produce clones of itself and asexually reproduce, to have lots and lots of little salps hanging off there in a tail until they're mature enough to break off and go swimming around by themselves. Hopefully we'll have enough to ID that guy now. That should be pretty good. So the yellow. Okay, that yellow spot's actually its stomach. It doesn't look to have eaten very much. It looks like there's an amphipod, that white, slightly longish structure on the surface of the animal. It's probably Verbelia. Thanks very much for that. That was good.
We'll carry on.
Yes. These salps are strange little animals. They're actually more closely related to humans than they are to jellyfish. Although they're transparent and soft, squishy, they have what amounts to a spinal cord. They don't have a backbone, but they do have what amounts to a spinal cord. So they're closer to the vertebrates and fish than they are to jellyfish. They swim through the water as the muscle bands around the body contract. It propels the animal through the water, and as the water passes through the body, the small food particles are caught on the feeding net and then ingested. Salps have a very high turnover of particles through their stomach, and they repackage these very small phytoplankton like cyanobacteria or small marine snow particles into much larger fecal pellets which sink at a much faster rate. So they're very important in the transport of, organic matter from the surface waters down into the deep sea. There's a very large object just went past. Sometimes what look like siphonophores when you're driving an ROV through the depths ends up being a squid that saw you coming and escaped while squirting out ink. And the ink becomes kind of a pseudomorph. It often takes up the same shape as the squid itself, so a visual predator will mistake the ink for the squid. And it sits there mixed in with mucus, and each pulse leaving out a little packet of ink, and the packets of the ink are normally about the same length and width as the squid that produced them. So it looks like there's all these little squids there, and the predator's going, which one is the squid that I want to eat? And meanwhile, the squid is either hiding somewhere in that ink, or it's hightailed it out some way to get away from being eaten. Many is the time I've said, oh, huge siphonophore, and come back and it's just squidding. I've even seen squids, after squirting the ink, come back and hover just below the ink, like hiding behind it. Because in the mid-water, there isn't any rocks to hide behind, there isn't seaweed to hide behind. So it's a continual fight for survival, trying to camouflage yourself in a place where there's nothing to hide behind. That's why so many of the animals are very transparent, and why jellyfish do so well. Especially because animals can come by and chomp a few pieces off a jellyfish and they just grow back. A good survival strategy. And there are some animals that remain permanently attached to the jellyfish, or remain permanently swimming around close to the jellyfish, chomping a bit off every now and again, waiting for the jellyfish to grow back and then chomping it off again. Are we able to increase either the lights or the gain or something on the camera, please?
Can you see okay, Dhugal?
It's still pretty dark from where I am. I am sitting outside, of course, so you probably can see things a lot more clearly than I can. It's good to have it slightly dark in the recorded video because we can bump it up later and we don't lose all the whites and the information at the very bright end of the spectrum. Just makes it a bit hard to ID the animals or see them when they're streaming. So I'll leave that to you to look at the video quality there. Are you actually able to make out the marine snow particles in the bottom right and bottom left corners?
Yeah, it's pretty clear here, Dougal.
Okay, let's leave it like this then.
Yeah, of course, it's quite dark in the control room.
Yeah. I just have the upper half and the central third is illuminated here. This looks like a mucus feeding structure. Yes, a mucus feeding structure. Some of these mucus particles, the very large ones, for example, the houses of Bathycordius lavations, these floating, filtering sea squirts, can be over a meter in diameter. And as these sink through the water column, they scavenge all the other marine particles around them, and therefore get heavier and heavier as they sink. They can sink at speeds of up to 1,000 meters a day, and by scavenging all those particles in between, deliver a very large food package to the benthic animals that live below. So for now, benthic scientists here, they're very interested in seeing what kind of food is being supplied to the benthos, and how much of it there is as well. So we're keeping an eye not just on the jellyfish and other animals living in the midwater, but having a look at the mucus and other things that are sinking through the water column, providing an organic rain of food for the animals beneath. As well as this continual rain of sinking particles, another important source of food for the benthos is the actively migrating animals. There's a worldwide daily migration of all sorts of little shrimp-like crustaceans and fish, possibly jellyfish as well, that head up into the surface waters at night when it's dark and they can't be seen and eaten by larger predators. And they chomp around on the little plant plankton and small animal plankton that live up at the surface. And then as it starts to get bright and they might be seen by their predators, they head back down into the deep where it's nice and cool. They can hang out there without using too much energy, waiting for the next migration at night. And the deep sea animals chomp them up if they can find them. The migrating animals are normally not moving around very much when they go back into the cold waters in the deep to digest their food during the day. So filtering animals are very good at picking those up, just continually filtering the water and finding these floating animals, things like gorgonians, corals, which have a large area that they're filtering the water through as it passes between their branches. A range of midwater animals that have very large finger-like appendages that they use to sweep through the water to try and catch any particles that are there. And then there's, oh, that was a jellyfish, possibly. But a ctenophore? I think I can see comb rose reflecting the lights. Is he still there?
Yeah, he's still there. They're just having trouble focusing in on him. A bit of movement.
Would you say it's at Singapore? I think I saw cameras.
Not at this end. It appears to be very transparent, Dougal.
Okay. Is it on the camera somewhere now?
Yeah, it's still visible.
See if I can increase my screen brightness a bit.
Almost coming into center screen now, Dougal.
Yeah, it's very, very transparent. That seems to be the problem.
Oh, there it is. Literally enters the top half of the screen, and I'll see if I can give you an idea. It looks to be pulsing, doesn't it?
Can you see it now, Dougal?
It's about to come into the screen. Oh, it's a Medusa of some sort. Not a ctenophore. Ooh. Ooh, that's Gerionia. Wait a minute, can I spell that? Geronia. It's a Trachymedusa. Most Trachymedusae have 8 radial canals. This guy, I think, has 6. You probably see 6 radial canals. And a very long, if you call it a proboscis, but the mouth is situated on the end of a very long gelatinous protuberance coming out of the bell, and a small white stomach attached to the end of it. So 6 very long tentacles, it looks like. So yeah, I have a positive ID for that. That's great. We have the associated temperature, salinity, depth, oxygen, etc. data. So we've got a hold on its environmental preferences. I know that species, and I'm fine with continuing on our survey now that we have that important data about what kind of environment this animal likes to live in. When we collect these with plankton nets, of course, for example, this animal here we see at 243 meters depth, we would only be able to say it lives somewhere between, say, 100 and 300 if we have a net that can sample discrete layers. Or if we have a regular plankton net, it lives somewhere between 0 and whatever the maximum depth was we deployed the net to. So it's not very fine scale, fine grained data in order to tell what environments these animals actually prefer. And if we hoping to have any grip on what's going to happen to the oceans as the climate is changing, as the warm surface layer becomes deeper and deeper, keeping the nutrients away from the surface by stopping the upwelling. If the salinity drops because of melting ice, all these different things that could be happening to the ocean, it's important to know what kinds of habitats and environmental parameters the animals that live there prefer, what they thrive in, what they can just survive, and if it gets to a certain point, if they are never seen there at all. So we're trying to collect this kind of fine-grained data in order to inform politicians and other people about what could happen in the future. It's also why it's very important that we're getting this data from off Australia. This area has very few ROV surveys having been done, and this is one of the first midwater ROV surveys that has been done off the coast of Australia, in the tropics especially. So this is very valuable data. pH values tend to be low around here, so it should inform us also about global ocean acidification related things as well. So heading down past 250 meters depth, normally one would expect to see krill kicking in somewhere around here. These krill are tiny little shrimp-like animals. You can tell them apart from shrimps because their gills are outside their body, in between their legs, rather than inside the carapace with normal shrimps. And they're an important food source to whales and to many other animals, including my friends, the jellyfish. One reason I'm so interested in jellyfish is not just because there's so many of them around, although that is very important, but there's a large number of species that occur in the deep sea. And unlike, say, birds, which have different mating calls to different birds, or they can sing, they can do different dances or make fancy nests, or some of them, like the cuckoo, can even invade another nest and throw out the eggs of the bird that should be there and replace it with its own eggs. They don't have all of this very interesting and developed behavior. They're jellyfish. They just kind of sit there or they swim. But the diversity is very, very high. So how did the diversity become so high, and how is it maintained? How can you have so many animals that have what looks to be a fairly similar lifestyle, all living in the same place and not outcompeting each other? I'm very interested in what this biodiversity maintenance mechanisms mean, not just for the jellyfish themselves, but if this can be applied to other systems as well. So what we're learning here in the deep sea, looking at jellyfish off Australia, could be applied to other places where we're interested in biodiversity, like rainforests, or other places where we can't quite explain why diversity is so high. The jellyfish also seem to be acting kind of like the trees in a rainforest by providing substrates for animals to attach to. You have a tree in the rainforest, you might have one insect that lives in the bark, one on the leaves, one in the roots, one tunnels in there. So you can have multiple insects, each dependent on the same tree, but different parts of the tree. We've actually found jellyfish with small shrimp-like animals, amphipods, attached to them, where one species was attached to the outer side of the umbrella and another was attached to the inner side of the umbrella near the gonads. And this was observed in multiple animals, this same living in different places, habitat segregation on the surface of these jellyfish. That's very interesting, and is maybe one reason why the diversity of the plankton is so high, even though they live in this seemingly homogeneous environment without seaweed and rocks and other things to increase the variations in the microenvironments as we move through. The ocean actually is usually very vertically segregated. It's kind of like a series of pancakes made of water all lying on top of each other, each with different characteristics. So in the vertical plane, as we pass through, sometimes we can see quite thin layers of different animals that prefer different environments occurring, but you can only get that fine-grained information and distribution by looking with cameras at how the animals are distributed. And it's necessary to get that information at the species level because each species has its own environmental preferences. That's why things like the high-resolution camera here on the SuBastian are so important for midwater surveys. Coming along to 350 meters depth. I still haven't seen any krill. Large piece of marine snow. Possibly a fish there. Yep, looks like a baby fish. It's got a backbone, so I can't tell what it is, but if we catch these things on video, we have a team of scientists, taxonomists around the world who will be able to access the images that we are capturing here, and they'll be able to chip in their dime's worth of, I think this is this, I think that is that, and share the information. To give us a more complete view of the community that lives here in the twilight zone.
Dougal, would you like to tell everyone about the special cameras that the ROV team and Dr. Andy Carroll, who's on the survey, have managed to attach to SuBastian to some of the special imagery that you're after?
Sure, I'd be delighted to. So while I'm over here in Japan, not actually being able to take part in the survey, Andy, whoa, wait a second. Top of the screen, what do we have here? That is very long. I think the head, the nectosome is the bottom left. From this distance, this looks like a siphonophore. Does it have two large gelatinous cylindrical bells there, or a series of small small bells, 10 or more? In the shape of the cyphosome, I think two.
Yeah, I could see two.
Okay, so this is that animal that can reach lengths of 40 meters, Preya. If we can zoom in on the head, so-called head, those two large opposed nectophores, to get a side-on view on both sides.
Yeah, the pilots are just having a little bit of a location issue, but trying to zoom in, Dougal.
Roger. So this guy has probably got hundreds and hundreds of stomachs, each one with its own tentacle. And it sits like floating here in the mid-water, hanging these tentacles down like a curtain of death, waiting to catch the krill as they swim through this like maze of three-dimensionally arrayed maze of tentacles on the way to the surface. And they eat and they're on their way back down again, and they have to pass through this curtain of death once again. Wow. So each one of those spots is something that'll be able to eat prey. And I guess I have the enviable job later of counting how many stomachs are on that. Doesn't look like many of them are full though. The Cyphosome seems a lot, the Chlamydia seem very far apart from each other. I'll have to look at those nectaphores later to decide if this is a rosacea or some other praeid rather than Praea, the genus itself. That's nice. Well, now I'll have to do that stomach count. Wow. Can you see embedded in the jelly any like very bright white bulbs or
Yeah, there's two.
structures? Okay, that should allow me to identify the animal. Ooh, Desmophes, Anectons? Yes, it's got two large globular white spheres embedded in the jelly there, hasn't it?
Yeah, that's correct.
Okay, so this animal is Desmophes anectans. Desmophys, anectans, Annectans. It's a praeid siphonophore, so we're in the right family, even looking from a distance. And the high quality video here has allowed us to tell what species it is. And I'll be counting those stomachs as well later. And we know exactly where it was living. So that's very good. Thank you for that. Nice data.
We right to go, Dougal?
Yep, that's fine. That's very nice. I don't think I've ever seen one with quite as many stomachs on it, this species. I never knew it could get quite so developed and mature. It just keeps, as it develops and ages, keeps adding chlamydia to the polyp colony. So gradually the numbers of stomachs increases until something swims past or runs into it, for example, and breaks the tail off, and it loses some of those zooids. Now those polyps, although they can no longer propel themselves through the water because they're missing the swimming bells, they can still survive for quite a long time, eating and reproducing as they slowly sink towards the sea floor and again provide food to the animals that live there. The main part of the animal that has the nectaphores on it, this is where the new polyps are budded off from, the area at the base of the nectaphores, and so it just keeps producing young chlamydia from there, and it gradually becomes longer and longer again. So this Desmophis anectans that we just saw must have been alive for quite a while because it was very long for that species. How you would actually go about aging a jellyfish, because they have no hard parts that you can section and polish and count the number of growth rings or anything like that. Quite a challenging scientific topic for someone who likes a challenge maybe more than me. Can probably get a bit of a handle on it by looking at the buildup of materials around the nerves. But it would be quite a challenging thing to try and find out how old a jellyfish is. But that is important data if you want to talk about their ecology and life history. We have another jellyfish coming in the center screen. No, possibly mucus. So I was starting to tell you about cameras, I believe. The SuBastian has its own array of cameras, including this 4K video camera that's on now, and some other cameras that are pointing in different directions. And this allows us to record the animals from like a front view and a top view at the same time. We also have several pressure housings rated to 3,000 meters depth, which Andy and our other colleagues there on the Falkor have attached to various parts on the ROV. There's the main one is a stereo camera system, which has two GoPros, which have been embedded in aluminium frames, and these frames are inserted into a larger aluminium pressure housing. This is to make sure they always face exactly the same way, so that we don't need to do multiple calibrations. Oh, wow, this is a siphonophore, possibly from the look of that, an apolemia, which is a very, very prehistoric basal siphonophore. And somewhere on one end of that spaghetti string, there should be some mostly transparent swimming bells. I think that's them there, yes. You can see some of the stomachs are larger, whiter, and hanging down. It looks like they've got krill inside them. So it's eaten at least a little bit. Quite a long animal. And if we can zoom in on that nectosome to look at the swimming bells, the number of tentacles that are coming out in between each swimming bell is important, and also whether they have patches of nematocysts or stinging cells on the swimming bells will give us a bit of an idea. This family, the Apolemidae, has a large number of undescribed species. So in order to actually tell what species it is, you'd have to collect one and put it together like a jigsaw puzzle afterwards. And I think this guy, though, would clog our suction sampler and might stop us from collecting other things. So we probably won't sample him. Collect a little bit more of this video.
What did you want to see in particular here, Dhugal?
I think top left screen just there, I think that's where the nectosome is. Can you see a group of biserially arranged semi-transparent swimming bells on the animal anywhere? It could be a ghost tail. The nectosome may have swum off and left this cyphosome to fend for itself if it was escaping from a predator or something. Or there could still be a nectosome attached. If the nectosome is attached, I'd like a zoomed in image of it, please. I can't see the smoking gun though.
Yeah, J-Rod thinks he's trying to zoom in on it now.
Right. It's definitely been eating a lot though. You can see all those things in the stomachs. Yeah, from what I can see in the video, it doesn't look like it's... Oh, that's it. Oh yeah, you've got it. That's the, in center screen now, you can see that bright white reflective thing is probably the float. And below that, all the nectophores are attached in 2 rows. And the cyphosome from there. So just, yeah, as close as in as you can get to that would be nice. You imagine catching this thing in a plankton net and then trying to put it all back together again to see what you thought it looked like? Awesome. Yeah, that should give me a good shot at telling what it is. Quite a large photo.
Shall we carry on?
Okay, thank you. Yes, thank you very much. These apolemias don't grow quite as long as the prayed siphonophores, which can reach a length of 40 meters for the colony. The largest animal, though, has been recorded at about 20 meters length. And I believe the Schmidt Ocean Institute survey off the west coast of Australia saw one of these apolemia last month or the month before, a very, very long apolemia siphonophore.
Yeah, J-Rod tells me it was over 100 meters.
Yeah, we got it at about 130 to 140 meters.
Holy mackerel.
The outer ring of that, when we went around, was the outer ring.
All right, well, we may have a new record.
Yeah, they're saying it had 5 or 6 rings, and the outer one was...
There's another long something. Rather than a cyphosome, that looks more like the tentacle of something that's broken off. These colonies of jellyfish reach the longest lengths, but some of the regular jellies can also reach very large lengths. We caught a jellyfish in Antarctica once back in 2008, I think it was, and it came up in a net that we were using to look for young pelagic cod and penguin food. And the jellyfish that came up there, I lay it out on the deck and lay next to it, and it was like 6 or 7 times my body length. And this one has very, very long tentacles as well. They do get swept further north. As we go deeper, we'll be heading into a water mass called the Antarctic Intermediate Water, which is coming from down near the polar convergence zone. Here we have something else coming into the video now. Looks like another siphonophore. It looks like Agalma okini, I would say. We get a bit closer, we might know. Yes, we saw one of these yesterday as well. At this distance, the cyphosome looks white, but as we get closer, the red, which is absorbed by the seawater, should start to become more and more visible. It should have like an orangish kind of a tinge to the stomachs and tentacles. So yes, I'd say that's Agalma okini, which is a siphonophore. Rather than becoming very, very long like the last siphonophore, apolemia, that we saw, this one tries to make its cyphosome as small and compact as possible. And it, the bracts, which are the protective zooids, interlock like Jenga blocks, and they protect the stomachs and the gonads inside from being preyed upon by other things that come along to peck at its zooids. That's great, thank you. I've got an idea on that. The stereo GoPro, which is attached to the SuBastian here, should allow us to tell the length of that colony, the diameter of the colony. And if we see groups of animals, like occurring in a swarm or a school, we should be able to measure the distances between each of those individuals to get an idea of how many of them there are in each cubic meter of water. Is that another siphonophore? Oh, this looks like Forscalia, perhaps? Yeah, let's take a look at this. Seems to have the tentacles are very widely spaced. They don't occur in a single line like in the other animals we've been seeing. Yes, I'd say that's a Forscalia. Rather than the nectophores being arranged biserially in 2 rows, they're arranged in kind of a spiral. So if it's a Forscalia and it swims forward, they normally turn as they swim. So you can tell the family of siphonophore from the way they swim. Our nickname for it is the Christmas tree siphonophore, because it looks like when you wrap all those glowing lights around your Christmas tree, the tentacles wrapped around the cyphosome kind of reminded me of that. So that's how it got its common name. So it's the genus Forscalia. There's several different species. We can see the attachment of the float to the nectosome to see if it protrudes or is pulled back in. Would be a good hint. And the form of the cyphosome from a side-on view will also give us a good idea. So again, compared to the Cairns seamount, this is it's a lot more siphonophores around. The biodiversity seems much higher. When you think you've got enough in focus shots at various angles, you can keep heading down. Oh yeah, that's good.
So we've got a couple of good side-on shots now, Dougal.
Yeah, I don't think it's Formosa. It's strange. The cyphosome is so spread out. Cool, thank you very much. We haven't seen any Nakamijusi yet this dive. And the salps are nowhere near as common as they were in the last couple of dives, especially the first one. So possibly there's the prey is more crustaceans or... Wow, what's that? What do we have here? Anyone want to take a guess? It's a bit dark for me to decide. Okay. Siphonophores. Ah. Is it possible there's a large supply of larvae from the neighboring coral reef that could be acting as a food source?
There's no surface reef nearby, but there's a lot of topographic relief on the seabed, so in the order of hundreds of meters within a very short distance. So there could be some quite active currents here, but apart from no surface reefs, but lots of deeper reefs. So a lot of mesophotic environment here, mesophotic seabed settings, where there's probably a lot of productivity there.
Okay. Is that a squid? Should look at that in the video later. Yeah, it just seems that the Cairns seamount looked to be mostly possibly like productivity upwelling, small plankton filter feeding, rain of detritus kind of supported, whereas a lot of these animals will be feeding on particulate, like small crustaceans, rather than filter feeding and going through the gelatinous web. There's probably less Narcomedusae around because there are fewer salps and tunicates for them to feed on. Good thing we collected them earlier in the cruise.
Now at a depth of 408 meters, and we're diving with ROV SuBastian on the steep northern margin of Malay Reef, Coral Sea Marine Park. This is the 7th ROV dive on RV Falkor, Mid-Ocean Institute's research vessel, and Seamounts, Canyons and Reef research cruise. Currently we're moving through the water column down to a depth around 630 meters. We'll meet the seabed, and after that we'll begin our ascent following geography of the seabed up to the surface waters. Joining us is Dr. Dhugal Lindsay. He's an expert in plankton, and we're observing and potentially collecting plankton species as we move down through the water column. Still got you, Dhugal?
Yes, you have. I'm staring at my screen here, looking in anticipation at our next animal that swims or floats into view. So many particles to kind of try and keep track of to decide which ones are interesting and which ones we can't tell what they are. I'm sure our ROV pilots there are also, as they're flying, because of the slight lag in time as to when I get to see the animals sitting here in Japan. Oh, that was a fish, Ceravoma perhaps? Looks like we've got a sawtooth eel maybe. It's just showing on the video. So they need to anticipate what it is I'll be yelling at in a few seconds time and already stop on it, or it'll be too late. So they're doing a good job here, like finding orcoplurid animals they think would be of interest to science, and stopping the ROV on them. Great big hunk of metal stopped on animals that can be like a centimeter in diameter, and swimming and floating around in three-dimensional space, trying to stop this huge ROV on them and get them in focus for long enough for us to tell what they are. And if we're very lucky, tell what they're doing down here. You can sometimes see animals attached to other animals, which when you sample with a plankton net, everything is all mixed in together, so you can't tell what was originally associated with what. Whereas with these in situ ROV surveys, we can say definitively these two animals occur together. Something we kind of take for granted with work we do on land or in the shallows, where we can actually see what's going on. So these ROVs are a very important tool for getting more of a handle on the midwater community. Ooh, that looks nice. Is that a single gelatinous bell or two?
Two, maybe. What do you think, Jared?
Two. If it's two, it's probably a prayed siphonophore. Looks like two. Different species to the one we just saw before. I think I can't see two globular projections into the jelly of the swimming bells.
Yeah, definitely two propulsion bells, if that's what you call them, Dougal.
Yes. Two nectophores. I wonder if this guy would be small enough to be able to suck it up.
Yep. Cody said yep.
This might be a good one to catch. I don't see anything before looking exactly like it.
Good to sample, Dougal?
Yep, that'd be great. And yes, So preserve most of this in formalin, and invariably a few pieces will fall off it, and a couple of pieces we put in ethanol to try and get a DNA barcode on it. This will also help us tell where it fits in the family tree of siphonophores in case it's an undescribed species. I haven't seen anything looking exactly like it before, so guess we'll have to wait till we get back to the lab to see exactly what it is.
Got him.
Got him? He's in there?
You think so?
Awesome. Excellent there. Looking forward to seeing that. Very cool. That's a Prayed siphonophore. A swimming colony of jellyfish.
We're just
All
checking the
attached to
jars.
those 2 swimming nectophores.
Yeah, so
Yep.
that's jar 2, is it? No, jar 1, sorry.
Jar 1. Praydidae. Praydidae.
Yeah. Yeah, he may not be in one piece anymore.
Yeah, siphonophores often like eject pieces of themselves because they can grow more back. You almost never catch them in one piece. Looks good. Very nice. So because I'm unable to actually take part physically in this survey, we have a team on the ship there. Marty, Jeremy, Andy, possibly some others also are pulling these animals out of the sample canisters after the ROV comes up, taking them into the lab and trying to photographically document their morphology. Because a lot of these jellyfish don't survive very well the preservation in formalin. So they're taking photos as well as they can before the animal starts to look the worse for wear, and then subsampling tissue for DNA barcoding and the rest of the animal, so that it can be observed under a microscope later for a positive ID and a possible species description if it turns out to be new. Wonder what we'll see next. Oh, that was a krill. He was donating. You can often tell the genus Euphausia because it swims in a donut with the back being in the central axis. And very few ctenophores on this dive. Almost lots of siphonophores. Good. Ooh, what do we have here? Speak of the devil. This looks like it might be Thalassocalus, a ctenophore. Comb jellyfish. Is it a Medusa-shaped ctenophore? I think I can see glittering comb rows there. The one we call the mask jellyfish because it looks like a rounded face mask with 2 eyes. Certainly looks, yep, that's the Thalassocalus. And Constans. Quite deep. Doesn't look like it's eaten anything. It's a great thing about jellyfish with transparent stomachs. You can take a look in and see if it's eaten something or not.
Shall we carry on?
Yes, please. Thank you. There you go. So just as I say, we haven't seen something, we see one. So let's try this. Well, we haven't seen a Solmissus yet this dive. And there's not many Trachymedusae apart from that Geryonia we saw before, which is also very interesting. So many siphonophores.
So we're approaching 470 meters. The water temperature is 11.5 degrees Celsius. And this is dive number 377 of ROV SuBastian.
We're operating everything in Greenwich time, Zulu time now. It's 25 minutes after midnight on the 10th of August in Greenwich time, which means I think it's 10:25 there in Australia. I'm here at 9:25 in the morning in Japan. In order to have all of our data synchronized, we work with universal time, Greenwich time. So we're trying to keep everything synchronized. Siphonophore. Krill. Surprised we haven't seen any more radiolarians or single-celled plankton with skeletons made of glass. A survey we did in the South Pacific, and there were over 50% of the animals we saw were these things that look like sea urchins. That looks like a cyclothone fish, perhaps? I think that's what that is. Maybe pteropod in the screen at the same time. This is cyclothone. It's actually the most common vertebrate on the planet. Most common thing with a backbone on the planet. Ooh! Is it a Sedipitenophore or a doliolid nurse? Sedipitenophore. And aha! Does this have the tentacle bulbs not lying in the same axis as the body, but actually at 90 degrees? Where those two long tentacles are coming out of the body. At the very base of them, there should be two bright white patches. We can see the shape of those.
There's definitely two.
Ooh, is this the undescribed species we've been calling Tetraphalia, perhaps? Be nice to get some good video of this. Oh, yes, I think it might be. It looks to be the comb jelly that we call the Japanese kite comb jelly. Because it's very flat, almost rectangular, I guess, but with long projections and two long tentacles hanging down as if they were the strings of a kite. certainly what it looks like. Very, very flattened body. Belongs to the most primitive of the ctenophore groups, allied to the Mertensid ctenophores. Oh, that's nice. Yeah, that's it. That's it, all right. Huh. So it is in the southern hemisphere as well. Very nice. Thank you. Oh, that's an interesting video. Very nice. Got a lot of different angles on it to tell the exact shape.
Very colourful.
Yeah, the combers are reflecting the lights of the ROV. Depending on exactly what angle they are and which row is interfering with which, they come back in a variety of rainbow colors. The jellyfish itself bioluminesces with a blue light. So any light that's other than blue you see on that animal is reflections from our lights of the ROV. It's very hard to study which animals bioluminesce in the deep sea because by the time you sample them and bring them back to the surface, they've often used up their stores of the bioluminescent substrate. So the enzyme can no longer act on it and it will no longer glow. So you really need cameras in situ to see the animals glowing while they're still alive before you sample them. Or you need very good animal husbandry techniques to keep the animal in a tank until it recovers enough to make more substrate. And in many of these animals, they actually get the substrate by through their prey. So they eat a copepod, for example, and they absorb the substrate the copepod has, and they use that to produce their own bioluminescence. Certainly a lot going on. There's a lot of organisms around. Definitely rethinking my assumption that this is an oceanic desert with spots of reefs, although we're close to reefs, so it could be because we're near a reef, there's so much action happening. So many of those marine snow particles look to be alive. So yes, back to the cameras that I started to tell you about, as well as the stereo camera, especially modified GoPros that are in a single stereo housing with a USB battery attached to give us a longer battery life and a very large micro SD card to try and collect hours and hours of footage. We have a couple of other high resolution GoPro cameras in their housings, one pointing forward and one pointing down. This gives us 3 different angles. They're very wide, widescreen images compared to the regular cameras on the SuBastian. So they give us the context in which the observations are being made. So while we're zooming in on this jellyfish, for example, the GoPro's fixed focus, fixed field of view, so we can get quantitative data on what's happening while we're zoomed in and like looking at this jellyfish. There may be a giant squid just off to our right that we're not seeing, and these other cameras will help us to capture that as well. And we'll be able to see the habitats that the animals are in when we're doing our benthic transects. If we're zoomed in on a crab or a sponge, we'll be able to look out and see just to the right is actually a little gorgonian, which is information you may not see when you're concentrating on one main video camera that you can pan and tilt. So it's a combination of different imaging devices that allows us to get a much better idea of the totality of the environment. In order to synchronize those offline cameras, it's necessary for someone who's actually on the ship there to be visible in the field of view of the camera and, for example, clap as you see in the movies. So we have a sound, something that we can go back to in the soundtrack to synchronize things. On all of the cameras that have microphones, and many of the cameras don't have microphones, so we catch things like that in the video track to synchronize those with the other cameras. And then we have a calibration board for the stereo camera. So you hold it at a variety of different angles, you rotate it, change the way it is, maybe turn it around, try and get a lot of different images of the calibration board in both of these cameras at the same time, and then we're able to quantify the space that they're in. Oh, here we have another midwater siphonophore, it looks like. Another siphonophore. Nectosome is to the left. Siphonosome, acatale, now above it. Maybe 30 chlamydia, maybe more. Siphonosome seems rigid. Oh, wow, it's fluorescent. Any idea? Is that green fluorescent glowing the
That's the tail, is it?
That's the tail, I think. Did it just lose its ejector? Oh no, the necrophores are still there. They're just very faint. Does it have two?
Two bells.
Yeah.
Yeah, we think so.
I'll put my money on it being a Lilioxus fluoracantha, perhaps, with that amount of fluorescence.
The bells.
Good look at those bells.
Yeah, J-Rod's confirmed, there's two.
Right. And there's no bulbous projection. In the jelly?
Today, score-wise, you know, since he's by himself, you know, I think he enjoyed the company. Yeah. Yeah, by himself since June. He said. Yeah. And he said it's like us. He's been married 35 years. So he said he can't imagine being alone. I asked him if he enjoys being alone. I mean, it's been hard. Yeah.
Yeah, we think we've lost it, Dougal. Shall we carry on?
Yep, that's cool. Thank you.
Right.
I think that was an animal. Well, I'll look at the video later on when I get the high quality stuff to give a definitive ID, but it looked to be a Liliopsis fluoracantha, just described by Steve Haddock. I think Phil Pugh as well, back in Monterey Bay several years back. I saw large numbers of them in the Indian Ocean off the coast of Mauritius many years ago. And so they're here in the Pacific in the southern hemisphere as well.
Oh, yeah, he's got two.
I see the altimeter has kicked in. We're 85, 90 meters off the bottom, perhaps, at 552 or 3 meters depth. We have another jelly in our sights.
Yeah.
It's a Medusa.
Where he's
It looks to have a round stomach. Does this look like the guy we caught on the first day? The Halotrophus? Rounded bowl-shaped bill, perhaps? Wide ribbon-like radial canals?
Australia. Because he's so alone down there, ne? He doesn't know anybody. This way, maybe. Yeah, Angus. Oh yeah,
Once you think we have a close-up in focus video you think I can get an ID from, we'll be fine to keep going. We have a sample if this is a Halotrophus, I think it is. Certainly looks like it.
Of course. Oh yeah. But 9 holes. But still.
I think I can see that the distal ends of the tentacles are a brighter white than the proximal ends, which is the family Halocreatidae, to which this probably belongs. Has these stiff rod-like distal ends to the tentacles. It's definitely another Halotrophus.
Every time we see this booklet with coupon, we've been collecting on those books.
Yeah, okay, that's good. Yeah.
So we have enough coupons.
That enough, Dougal?
Yep, that's great.
Okay, let's moving.
50 cents. Got a job?
Did she find the job yet? No.
Almost at 550 meters depth.
Yeah.
Still only a couple of ctenophores. Many siphonophores. elevation house, double lobed.
Yeah.
Oikoplurid elevation house. The filter feeders are quite deep.
It's a beautiful
Possibly
day.
there's a krill, I think. What's that? A little siphonophore. Dified, perhaps. Does it have a rocket-shaped bell? Does it have two bells joined together to form a rocket shape? In which case it's a dified siphonophore. No. Wait. Is it a tomoctrid? Is that a polychaete? I have to check on the high quality video later. Like a fluid house. More filter feeders deeper, possibly there's resuspended sediment or like a lateral flow of matter from a ledge or something on the seafloor out to here.
Hello, everyone. Jeremy here. I just wanted to answer a few questions while we had a moment of pause. The target depth for today will be about 600 meters, and we're nearing that depth now. We're at 560 meters. And just one more question I saw on the chat, how many cameras does ROV SuBastian have? It's 9 cameras and additional cameras for Dhugal's work. Dhugal, how many cameras that are yours for your research?
I've got 3 camera housings. 2 have, whoa, soul misses! 2 have one camera each in them, and 1 has a stereo pair.
So every square inch of the RV is being viewed.
Covered in cameras. Yes. The soul missus looks to have eaten something. We can see something in its stomach. It looks like the White Sox morph. And after getting some video, it would be great if we could suck him up.
Certainly.
Dinner plate jelly. Very, very important top-level predator in the midwater, feeding mostly on gelatinous animals. We did record one a year or 2 ago. Oh, nice stomach pouches. that had a fish in its gut, but it may be that the fish had been eaten by a jellyfish, and then Solmissus ate the jellyfish and ingested the fish, therefore, at the same time. So a bit of a bonus snack that came along with its main meal.
I'll tell you, so Marty and I are photographing all of these jellies that we're collecting, and it is so difficult to image these things with, to get them in focus. So we've done what you said. We've tried putting the light on the side, shooting it through a petri dish that's elevated. Man, is it difficult.
And autofocus doesn't work. You have to have fixed focus and go slightly forward and backward, click, click, and hope you get it. It's very hard on a moving ship, even where the jelly in the petri dish is moving as well.
Yeah, we're kind of hoping you get really good images from this, from what you see in situ, and once in a while,
looking nice.
Ones with more features like this one, I think we could focus on the outer bit. It's got more features to see under the microscope, or under the camera.
Yep.
Yeah, let's collect it.
Thank you. This will probably be another pizza slice collection. I'd imagine its diameter is too big to fit in a suction sampler tube. The jelly won't be, if it doesn't fit in and we only get a piece, the jelly will grow back. They have very high regenerative capabilities. Oh, it might fit. Well, that was quick. Very smooth. Excellent. Oh, and there's an amphipod, so we must be near the seafloor. The first jellyfish we caught today, Jeremy, is a Period siphonophore, which probably will be in several different pieces because it's a polyp colony. So each of those pieces can kind of survive by itself without being attached to anything else for a certain amount of time. Oh, it's another Solmissus. Wow, they're kicking in right near the bottom. We didn't see any in the water column above here. Does it look to be the same morphotype? It's got tentacles. Cool. Perhaps more of the food they like deeper.
So, with the mid-water work, using the lasers usually isn't really helpful because it'd be shooting it out to black in the backgrounds. But with a really large jelly, maybe like this one, it might actually work. I think Dhugal.
Lasers, you mean?
Yeah, to 10 centimeter.
The light, you could have a go. The light normally is very penetrative and it doesn't reflect back very much, especially because they're normally green lasers. But yeah, normally you get, even if it's hitting the jelly, and it's really hard to hit the jelly in 3D space,
Right.
you may not actually see them.
Yeah, we're trying it
where
now.
they hit the jelly.
There you go. Okay, yeah. Working for a moment. You'll get a you'll get a snapshot in there where you see both lasers hitting the jelly.
Wow. Great. Awesome. That'll be an extra check for the stereo GoPro cameras to make sure they're actually getting the correct sizes. So that'll be very good. Better make a note of that, maybe.
Thank you, Salma, for the suggestion.
Awesome. Can we suck this guy up as well? And looks like we're getting good video of it.
Yeah, let's give it a try.
We're trying to do some biogeographic work and 3D habitat mapping using this Solmisus genus right now. So this is very important data we're collecting here off the coast of Australia in an area for which we have no data at all about Solmisus distributions or types, species. Every day, pushing back the boundaries of knowledge. Oh, this one's a bit bigger. Oh, got him. Did he go in the jar? Looks like it. Very nice. Thank you.
I'll just answer a question on Facebook. How do you do your sampling? So with the jellies, we take 1 or 2 tentacles and put it in ethanol, 100%. That'll be used for molecular analyses. And then the rest of the sample is put in formalin to preserve the structure of the jelly. I'll go to Dhugal and he'll spend, I'm assuming, many, many hours looking at our the jelly samples and determining what they are and what new information it brings to the field of jelly taxonomy.
And a lot of these animals were described in the latter half of the 1800s.
Yeah.
In archaic German or other languages. So we go, it's a real mismatch where we go through this really high technology ROV collected and like imaging in situ in the deep sea specimens, and we have to correlate them to the
literature.
These manuscripts written back in the 1700s and 1800s to tell on damaged specimens coming up in nets to tell what they are. Actually, back around the turn of the century, they used to use silk nets. So these were much better for collecting jellyfish than the nylon nets that we use today. They didn't damage the jellyfish as much. So we actually have a lot better data from back at the turn of the century. Whoa, big lobe ctenophore. Possibly Keohimea. We look to see whether it has a flattened body, triangular shaped, with two little rabbit ears sticking out near its backside. And then whether there are filaments attached to those little triangular projections or not. The filaments or not will tell us the genus, and the triangular shape and the flattened body and the very long ribbon-like auricles, 4 of them on either side of the lobes, will tell us. Oh, it looks like it's got projections, doesn't it? It's the family. I don't think I saw filaments there. Let's say that was probably Keohimea usagi. This particular species, if it is Keohimea, can grow very large, 1 meter or so in length. Catches its prey, its entire body surface is covered with a cell type called a coloblast, which is very, very sticky. So it's like swimming around with your body coated in fly paper. And it tries to catch all these small crustaceans, possibly even eats marine snow, sticking them to its body and then using cilia and its tentacles to move them to the mouth. Can you confirm, Jeremy, that there are no filaments coming out of the ends of the triangular projections near the anus?
I don't think so.
Okay, so we have Keohimea usagi. Yeah, I can see the tentacle bulbs flanking the mouth there as well. That's good. Thank you.
So collection or move on?
No, move on.
Right.
It won't survive preservation in formalin, and I think it's too big even to collect the entire animal. Need to keep. Oh, there's another Narcomedusa. Looks to be a Solmesis. Has it had its tentacles eaten by something?
The tentacles are faded. Do you want to look at it closely, or?
Just come in close and get an in focus shot.
See,
So it has very few tentacles, it seems.
7 tentacles.
7?
So, well, it looks like there's 3 on each side. No. Okay, 8.
8? Really?
There's 2 small ones on one end, separate from tentacles on each side.
And it doesn't really look like they've been chomped off, does it? Any of the others? That's the original number.
Yeah, seems to be consistent. You know, each section, if you put it into quarters, there's 1 quarter is 3, 1 quarter is 3, 1 quarter is 2, and the other has none.
Wow. So that's weird. we maybe suck this up and then I'll call it a day and let you get to your seafloor survey. I don't think I've ever seen an 8-tentacle Solmistus. Can you make out whether it has stomach pouches?
I'll go out on a limb and say no.
Cool. We may have a missing link. I think it's got structures on the rim as well.
If it's a new species, Orwitzi would be a nice, it's got a nice ring to it.
Yeah, there you go. There you go. It looks like something might have chomped at it once and it's grown back.
Could that modify what we're seeing now? No. The fact that it's been bitten and kind of looks different than it did.
Where those two really short tentacles are is a bit that's grown back, and on the other side as well, it looks like it's been chomped and is growing back as well. Maybe. We should know when we look at the specimen though. Quite small. Different area, isn't it?
So, yeah. I guess we could look at this one as well. Would you like to?
That looks to be the white socks morph. Did we actually sample that one we just tried for?
Got it.
Yep. Excellent. Thank you. And this is a more regular one, like the one we just sampled before, it looks like. Yeah, okay. That's good.
All right.
That's for gonads too. And stomach pouches. Thank you. $570 meters depth, about 50 meters above the seafloor. Slowly heading down, doing an oblique midwater transect, looking at the midwater community on the way to the seafloor. Lots of animals floating around here. Ooh.
That one looks like it's got very long tentacles.
Yes, maybe a machacomid. Not Colobanium, something else. First time we've seen this on this cruise. It also looks like it might have been chomped. It looks like it's only got 3 radial canals left. Yeah. So even if it was a new species, we wouldn't be able to describe it because it's not in the original morphology. It looks to be mutated or growing back. Be nice to find one of those. It's got 4 radial canals on one of our subsequent dives. Thank you. That's a Colobanium, I think.
Dougal, there's a question about bioluminescence among jellies.
The jellyfish on your screen right now is highly bioluminescent. Colobanium sericium. You'll notice the tentacles. It has a massive tentacles of different lengths. What this jellyfish does is when a predator tries to eat it, it ejects some of the tentacles, just like a lizard drops its tail, and uses it as a decoy. And those tentacles will glow in the dark. And the fish, or whatever predator it is, will go after the glowing tentacles while the Colobanium medusa swims away and escapes. And it does this repeatedly every time something tries to eat it. So as the tentacles grow back, they occur in a variety of different lengths. You can see how many groups of different lengths tentacles they have, and have an idea how many times it's almost been eaten in the past. Some jellyfish produce their own bioluminescent substrate. Others get that substrate from their prey. So if you have a jellyfish that normally is eating like copepods or ostracods, something that has this bioluminescent substrate inside it, it'll be glowing when you catch it, and you keep it in an aquarium feeding it brine shrimp or something, and it won't glow again. But then you feed it copepods caught in the deep sea, and it'll start to glow again. Many, if not most, of the animals in the midwater can emit light. And even those that don't, they still rely on light to find their prey or to find a mate. This jellyfish is fairly easily recognized because as it reflects light from the ROV's cameras, the tentacles have a kind of bluish hue to them, which is definitive for the species. So even from quite a distance away, you can see it. You may be able to see the stomach is hanging down in the bell there, about halfway down the bell. Normally it would hang in the middle of the bell. It looks to be lying up against the inner surface right now, with a very long and narrow stomach.
There's a question about do jellies have organs to sense up and down?
Yes, the jellyfish have things called statocysts, which are usually placed in between the tentacles on the rim of the bell. There's a solid concretion. I think it's probably magnesium. There's some calcium carbonate or maybe magnesium carbonate that they use there. It differs in different groups of jellies. And it's like a solid rock, which is resting on some hairs, some cilia. And depending on which way the jellyfish is sitting, the cilia that are supporting that little statocyst, statolith, will bend, and they'll be able to sense which way is up. There are some jellies that no matter where we do our surveys, whether any distributing based on temperature, salinity, or light levels, but rather on the actual depth that they're at. So it seems they can sense pressure as well. But don't ask me how. Okay, Okay, thanks very much for that. That's some great footage. Colobanema sericium, the rainbow jelly in Japanese, because when you shine light at it from the side, the musculature tends to disperse the light in like a rainbow pattern. Very nice. This jellyfish is a hydrozoan jellyfish. The hydrozoa are the most specious or diverse of all the jellyfish groups living in the ocean. The jellyfish we're used to seeing are the meaty, solid scyphozoans that we normally see down at the beach. The hydrozoans are normally much smaller or more transparent, but there are actually more of them, and there's more different kinds of them as well. Some of them can occur as huge polyp colonies that can float or swim in the water, reaching lengths of many tens of meters. There's a fish there. And some of them are so small that they live as parasites inside the gonads of freshwater sturgeon paddlefish. You can also get freshwater jellyfish that live in lakes, so they're pretty much everywhere. There's many, many different types. That looks like a salp dragging behind it clones of itself in like a tail. Salp is, although it's transparent and gelatinous, is not a jelly, not a true jelly. It doesn't have stinging cells. Instead, it has a spinal cord and is actually closer to fish or humans than it is to a jellyfish taxonomically. They're very good at filtering the water and taking the food out of it, and growing in huge population bursts when there's high productivity in the surface waters. Looks like we've hit the seafloor.
Okay, Dougal, I think, yes, looks like the seabed's actually being mobilised by bottom currents at some stage. And we've got quite a sandy seabed with bouldery rocks here and there.
Thank you very much for a very productive midwater survey again today.
Thank you, Dougal. I think all the people viewing this on the internet have really enjoyed your commentary and appreciated the huge knowledge you've got of these jellies, and it's really been educational for me as a marine geologist to learn a lot more about the biological oceanography of just of the Coral Sea Marine Park, but the global context you put it in. Yeah, that was really nice.
Okay, thank you very much, everyone, and great appreciation to the expert piloting skills and collection, and my commiserations for the people who will be helping work on the jellyfish specimens, because they're so hard to photograph. Jeremy and Marty and Andy, and thank you very much for making all of this happen. It's a pity I can't be there because of the COVID pandemic, not allowed to travel, but it's great to be able to take part in this survey in real time here from Japan.
Okay, thanks again, Dougal.
Okay, hope you have a good rest of survey.
Thanks very much. I think we're just about on our beginning of our ascent. We fire off our waypoint, Jarrod. Okay, it's great. So we're just about to start our ascent back up the steep northern margin of Malay Reef. And you can see we've got a big shell on the seabed that's colonised by a few. Today we're making our way back up the northern margin of Malay Reef. This is the 7th dive of the Seamounts, Canyons and Reefs research cruise on ROV Falkor, the Schmidt Ocean Institute's research vessel. On the way back up the steep margin will be the biological communities and the sediments that we can see, physically sampling some of the biological communities of interest, in particular corals and sponges. And we're also very interested in sediments that are moving down the flank of the seamount, interested in rocks that are forming the flanks, and we're looking at the geomorphic features on the flanks. As we've seen in previous dives, there's quite a variety of deposits and erosional and depositional features on the flanks of the seamounts, and they're giving us some really good insights into major driving processes that are shaping the sea, shaping the reefs, and also creating some of the habitats that the species of interest are growing on. Now sitting at about 600 meters below seabed, the water temperature is 7.6 degrees Celsius. And I'm just about to hand over to our water coral expert, Jeremy Horowitz from James Cook University. Jeremy is in particular interested in black corals. It's the subject of his PhD, and been very successful in locating some of the genus and species of interest to Jeremy as part of his research. Just tuning the cameras to get a shot of this shell. And it's a small stony coral growing on it. Is that right, Jeremy? Possibly. Yeah, okay. Over to you, Jeremy.
Sure, let's bring it.
Okay, hello everyone. Yeah, we're just gonna get in a stable position, take a nice zoomed in image of the coral, and then we'll collect it. So at this depth, of course, you're going to get, if it's scleractinian coral, it'll be a zooxandelate, which means, it doesn't rely on sunlight as a source of energy. But there's also things that look like hard coral, like scleractinian hard corals, but they're not. Some hydrozoans are very clever in making me think they're hard corals. Yesterday we collected a stalastrid, which I initially thought was a hard coral, but I was then scolded by my supervisor, Tom Bridge, because they're lace corals. So we're just going to quickly focus in. I got it.
You got it? I think you're going the wrong way. There you go.
All right, and then we'll just zoom in a little bit, then focus a little more. You can see the tentacles. There we go. All right. So what we'll do now is we will collect it, and then back on the deck we'll take better images with our cameras and our USB microscope, and we'll consult the experts and figure out what it is that's sitting in the sand at 600 meters. So we, on the beginnings of these dives, when we, hit sand, we often find corals. And usually they're attached to things like shells. Like yesterday we found an amazing black coral on a nautilus shell. So what this tells us is that these corals need some sort of hard sediment to grow. So when a nautilus dies and those air compartments are somehow broken to allow water to get inside, it'll sink. Yeah, A is fine. Or actually, why don't we put it in E and box 2 and leave A, B, C, D for rocks. So yeah, shells that reach the sea floor provide suitable substrate for corals to grow on. Yeah, I don't know what shell, some sort of a conflict shell.
Good look at it?
Nah, it's okay. We'll get a look later. But there's probably lots of little other inverts growing on the shell. So we'll take a good look at the inside of the shell and the surface. Very good. All right off the bat.
Are we doing a core?
Yes, please. So now we're going to do a core sample. We're going to sample the sediment. And if you've been watching over the last few dives, the sediment here, it's quite, the particles are quite small, and it presents a challenge for us to suck up a sediment sample. So often what they'll first try to do a proper core sample. If that doesn't work, they'll come in from the side and scoop it up. Something's better than nothing.
You want stuff for himself. Slide. Oh, right on the job. Anywhere?
Yeah, anyway, it's good. Thanks, Becky.
It's very saturated, so. It's pretty hard there. It's probably some,
If you get past it, it'll actually hold.
some big chunks in there. Yeah.
There you go.
Yes.
You're about to bottom out.
So potentially the coral that we just collected was another stylastrid, but we'll confirm back on deck.
You're just pouring it over.
Very wet, so.
I think you got deep enough to make this a good sample. Correct.
No! The top real quick.
One thing would be short, the springs don't have enough tension to hold them on.
They were working all right.
South.
Yeah, I saw it kind of churn up when we first started.
Happy with that, or do you want it cleaner? That's pretty good.
That's pretty good.
Okay.
Yep.
Yeah, sorry, YouTube and Facebook. I don't think anyone here in the room, in the operation room, is a singer. Anyone want to sing Push It?
What, is that Rachel? Did Rachel asking for it? That's Rachel's job.
Really?
That looks.
I'm bringing the porch out more to you.
Maybe? Yeah. All right. Yeah, we got it. We got it.
Go, go, go. Yeah, talk is still good.
Yeah.
Nice.
Oh, y'all just want the top? Yeah. Oh,
As much as we get,
Well, yeah.
whatever we get, it's good.
top's, yeah, important.
Hey, I'll give you a B+ for that. It's pretty good.
Thank you.
You want another one or just one? There's enough. One's enough.
One was enough.
Yeah.
Maybe we can get Narita to call in and do some singing for all of you.
And then,
It's upwards.
how's that? You want to bring it in just a little bit closer?
Yeah.
Just kind of dust off a little bit.
Yeah, I mean more there's enough of them growing more or less together that they get, they fuse through microbial sediment gets trapped in them and they keep growing. And you don't want
so that's not a solitary column, that's a coral column.
Yes.
Like you don't have a shell or a interested lots of them occurring together to make a structure or grow, change the structure.
Yeah.
What's that?
All right, so successful coral grab and successful push core, and now we're off. So we're looking for, so on this expedition, we're looking for soft corals, hard corals, black corals, and sponges. And within each of these groups, you'll find different species that are found in the deep and ones that are found in the shallow. So in the deep, you can get zooxanthellate hard corals, which potentially is what we just collected. And then you get zooxanthellate hard corals in the shallows. And Tom Bridge at the Museum of Tropical Queensland is interested in sampling hard corals from both the shallow and the deep to better understand the evolutionary history of the group. Similar with the soft corals, with Andrea Quattrini and Kathy McFadden, who are interested in the same question with their specific group. And the same with the black corals. We're interested in the deep and shallow black coral species. Understanding how they are different between deep and shallow will help us understand how the different groups have evolved. So it seems that there's a really deep divergence between the deep black corals and the shallow black corals. When you look at a phylogenetic tree or a depiction of how the different species have evolved, you could see that a very long time ago, about 250 million years ago, the deep and the shallow lineages split.
Awesome, you're at a good.
So that means the shallow water black corals are more similar to each other, and the deep water black corals are more similar to each other, to themselves. And so that depth separation can relate to their molecular relationships, which is pretty interesting. And that hasn't really been discussed much in literature, but we are now learning this, and so this is something we're going to try to better understand through collections during this expedition. Specifically, collecting specimens that look very similar, but are both in the deep and the shallow, which will tell us that potentially convergent evolution is at play, where although they look the same, they only look the same because the habitat, the abiotic characteristics are pushing these corals to evolve in a certain way, whether it's unbranched or they're branched in a specific morphology. So these are really fundamental yet really interesting questions that we can now answer because we have advanced molecular techniques. So all of us, so with the soft corals, the black corals, and the hard corals, we're all using a molecular technique known as conserved elements. And so it's providing us with a lot of information and very results that have very high, what we call bootstrap values, or very high confidence in the depicted relationships between the different species. So for the first time ever, we're very confident that what we're seeing depicted in these phylogenetic trees represent the true relationships between species in these different groups. So it's really exciting. Now we just need lots of specimens. Here we have a fish.
Or is it too small?
Way too small. And currently like Sasquatch, out of focus.
This could be a ponyfish. Thank you, Dougal, for the ID.
Oh, he's good. He's good. So far.
So there's a question on YouTube about the sediment. Is this all coral rubble? So I'm going to assume that a lot of this is coral rubble and sand and shells. Essentially things that die in the midwater or on the reef will tumble down to this plateau and become sediment for new life. So there's definitely coral rubble here. Also rocks.
So Jeremy might mention the lasers.
So we have the lasers on now. They are 10 centimeters apart. So you can kind of see the size of the different fragments. Most of what we're seeing is much smaller than 10 centimeters. The fragments could be 2 to 1 to less than 1. So we're looking across the seafloor, and this will allow us to find corals, little corals that are sort of cryptic, sort of hiding in the sediment. They could be easy to miss, but when you do find them, they often represent really interesting specimens. So everyone keep your eyes peeled. A question on Facebook about finding meteorites underwater. I haven't. You guys ever find anything that looks like a meteorite?
We went on a search for them we had NASA on board where they do unatlantic. And we tried to find them, but they're going to look like rocks, so we had to sift through them all. We don't know. They think they had some, but they had to take them back to the lab. We never heard.
They never told you, huh?
We never got confirmation.
Probably alien life or something. So there's a question about sediment samples. I'll get back to that in a second. Can we just slow down near the rock here?
You may just sit down for handovers then?
Sure. Can we? So maybe in here?
This is where we're starting the 500-meter transect, and this is where we're starting just to learn
Oh, the whole time is fine.
this way. This way. To about right here.
So we found 3 rocks, large rocks in the sand, which, as you could see, are providing sediment for corals to grow on. And so while the ROV shifts, we're gonna see what we have here. Seems like soft corals, but we'll take a closer look. Thanks. Okay, we're just going to zoom in.
Oh, no.
This. So we got a crab. And so this doesn't. Yeah, we're gonna try to look at a closer. Beautiful crab, indeed. So we're gonna look at a slightly closer specimen. Can we turn autofocus off?
It should be off now. Yeah. Sometimes I get a sync between your controller and your. Does yours say it's on or off?
It says autos. It's on now. That should be off.
That's over there now. Yeah.
There we go. Got it. Thanks. Oh, sorry. All right.
It's better.
It's better. Yeah, it's in focus. All right. While we think about what this is, let's look at the soft coral to the left. So this might be a bamboo coral or another type of soft coral. Yep. We'll just take a nice image of this, then we'll collect this guy. We should get the lighting right.
That's ours.
Thanks. And it's, yeah, beautiful. There we go. Okay, so it's certainly a soft coral. I'm just going to zoom out and get a laser on a branch, and then we'll collect it.
You come here. No, you're good. 2 hours.
Okay, that'll do. Wait, it's just.
You okay? Is that good? Yeah, it's fine.
Yeah.
Sit here, critique you guys.
That'll do. All right. We'll grab it.
Okay, so. Might just reposition this a little bit.
Yeah. Oh, there's a mantis shrimp here.
I'm going to verbalize all of our things. Can you go back and look at that one?
Or some sort of crustacean. Yeah.
What are we getting?
Hello, you. All right.
But I got to reposition.
Yeah. Go ahead.
Happy?
Yeah. So we're probably going to also collect that guy there. So if you want to be right here, you can grab that.
Okay. I'm just going to stay a camera.
It's all yours. Thanks. Yeah, thank you. There, to answer your question, Lisa, while we're collecting, there isn't, well, of course there's a date and time stamp on our video footage, It's just not what you're seeing.
How much of a, just grab some water. How much of a piece do you want here, Jeremy?
Grab it near the base and you'll probably get a few.
You want all of it?
No, I don't really need all of it. So if you can take one branch, that's fine. If you can pick off a branch.
Let's take this down.
Maybe like this outer branch here.
Beautiful with the yellow dots on the screen. That's nice.
Jump over here.
Yeah. Yeah.
How's this, Ben?
Beautiful. Thank you, Ben. You can put it in E. Box 2.
Box 2, Ben.
So he loved that. The ROV live
Yeah.
flying in it. So can we record that?
So number 1 is copied. Now you want to get that little white guy in the back there too.
Yeah.
Go get some tentacles.
Yeah. You'll probably have to grab the whole. I'll zoom in for you. Oh yeah, you can see tentacles coming out.
Yeah. This will be
Oh no, those might be
super.
anemones growing on the coral.
Yeah. So
Yeah. So I don't see tentacles coming out of the.
So these guys are hard. So that's a hard square to coral. Yeah, there might be some others going on, but
Is this soft?
It should be hard.
hard. Yeah.
Yeah. So we'll probably pop the whole thing off. It'll probably just be easier than grabbing a coral fragment.
Range is going to be super delicate. So a little bit to come off. So I guess maybe coming in from into that base or kind of old parts of the to this substrate. Give it a shot.
Just use mints.
That'll do. Okay, yeah, we're not sure what it is, but it certainly looks interesting. You want to scoop it?
Already fed off.
You want to scoop it up?
Yeah, but it's
I mean, it's alright to crush some of the branch edges, as long as a few remain, which they will. It's alright to kind of chomp down on it and throw it in the bio.
nice.
Yeah, that works. There we go. So you can see how brittle this coral is. So to identify what it is, all we really need, well, we have images of how it branches. Can you put it in BioBox 1A? So it's super brittle, but if we have, and it seems like we do have a viable, like a complete branch that wasn't crushed, we'll be able to use the features on that branch to identify what it is.
It suction most of it.
Oh, that was great.
You want to suction most of it?
Oh no, it's okay. No, we got enough. All right, I think we sampled all that we want to in this spot, so we're gonna levitate and head forward. So I just wanted to take this time, as we're looking for more things to sample, to explain why we're sampling. So the purpose of this expedition, among other things, is to understand what biodiversity, what species live in these habitats. So this is one of the first times an ROV is sampling in the deep coral sea. And it provides us the opportunity to do long transects and sample the species that are in these different habitats. And what this information will tell us is how many species live in the different reefs that we're sampling, how common or rare the species are. And it also tells us more about the evolutionary history of these groups. But in terms of conservation, biodiversity underpins all conservation decisions. Oh, that's a big anemone. That's huge. So if we don't know what species live in these habitats, we have no idea about how to go about conserving these different habitats. You know, you have a limited amount of conservation resources. You can only protect a certain area of an ocean environment. The only way to make an informed decision about where to protect and where to leave open for fishing and other activities is to first sample the biodiversity, figure out what's living there, and then use that information to say, okay, we're gonna preserve this area because it has many rare species, there are this many threats towards this species, and therefore protecting this area will lead to a high conservation impact. So collecting samples now, and of course with permission from Parks Australia, and the Great Barrier Marine Park Authority. We're going to allow decision makers to make more informed conservation decisions right after we're done with this expedition. I see some talk on YouTube about you guys not being experts. I'll tell you now, you guys are identifying things that I can't identify. So, in my eyes, you guys are the experts. So keep watching, keep studying, keep reading and learning. That's how any of us get into it anyways. It's a good question on YouTube about potentially collecting rare species. The interesting thing about the deep sea... Oh wait, we're gonna slow down here. We're gonna stop in front of that coral.
Roger, thanks, Phil.
I'll respond to that question in a second. Let me just see what we got here. So it looks to be a sea pen, soft coral. We've collected a few of them so far. I'll just take a close look quickly and then we can move on. Yeah, looks like a sea pen. Okay. Yeah, that'll be good enough. All right, we continue. Thanks. So, going back to the question about potentially collecting a really rare species. You know, what if the sample we collect turns out to be the only one of its species? The interesting thing about the deep sea is that it consists of the largest area of the ocean. So, you know, something like 90%, or sorry if the percentage is a little off, but most of the deep sea, most of the ocean consists of deep sea. And the conditions in the deep sea are fairly similar. The temperature tends to be really cold. Right now we're at 8 degrees Celsius. The deeper you get, the colder it goes. There is no sunlight. So these suite of abiotic features allow for certain species to grow and survive and reproduce. So if a species can live right here, there's no reason why it can't live elsewhere. And by that I mean, you know, it's unlikely that the specimen that we found is the only one of its kind. If we see it, it's likely to be common elsewhere in the deep sea. And this is different from, you know, a species occurring on one mountaintop where it's a very small area, and therefore any rare species that live there are there won't be many of them, right? So if you find a species on a mountaintop where the surface area is very small, just, you know, a few meters by a few meters, you probably wouldn't want to grab it and sample it, at least not without first making sure there's others elsewhere. But there has been a lot of exploration in the deep sea, and it seems like species in the deep have much wider geographic ranges than species in the shallows. And by that I mean many species in the deep are considered cosmopolitan, or occurring all over the world. So the deeper you go, generally, the larger the range of this species is, which means there's more of it found elsewhere. Can we just quickly look at that as well? Probably a sea pen. You don't even need to stop, I'll just zoom in. So we're just going over what probably is another sea pen. Okay, yep, sea pen, keep going. Okay, we're going to start picking it up a little bit. And so we're currently at 542 meters. It's 9 degrees Celsius. So our target for today is to sample at all depths. So from the deep, which today means 600 meters, to as shallow as we can go.
Ours is the summer.
Maybe there's something here. So we're coming across what looks like, yeah, there's a rock with some more soft corals, it looks like. So we'll just zoom over the top of this rock and quickly peer down to see if there's anything. Oh wow, look at all the big anemone. Oh, and there's a crab next to the anemone. Let's quickly. Want to drop down for a second, or we?
Yeah.
Just take a good look at that anemone there. All right, we're just gonna settle down for 1 second just to see. It looked like a big crustacean next to this anemone.
You look down, should be right for there.
There's the anemone. And try to zoom in before the sediment takes over. I think that's him in the center of the screen, but he's not moving. Which is very clever, because we're not going to wait all day. All right, we can move on. So there's a question on YouTube about the yellow, the polyps on the bamboo coral. So they have, they're soft corals, which means they have a polyp, a feeding mouth, and tentacles. And so the tissue has color, and so the color of that specific colony was yellow. I'm not a soft coral expert, so I'll leave my answer at that. But we can have other experts dial in over the next few weeks. So maybe we can get a bamboo person on the line. See some things, we'll just pass over that, I guess. Yeah. Okay, we're seeing more soft corals. Okay, I'll keep going. There's a shell with something living in it. Yep, looked like a snail. Oh, sorry. The next snail we come by, we could stop and take a closer view. Sorry, if we spend too much time looking at all the interesting stuff, we have less time for reaching the shallow waters. And if you'll notice on the previous dives, we always seem to run out of time towards the end of the dive, which always ends up being the shallowest portion, which means we're sampling less in the shallows than we would like. But today is a nice long dive, so we should have time to do a few stopovers. I'm going to quickly grab some food, and I'm going to pass off the mic to Brendan Brooke. Here you go, Brendan.
Thanks, Jeremy. As we continue our dive up the steep northern flank of this reef structure, this large platform reef, as you can see, there's lots of sediment being moved down the slope of Malay Reef, and with these occasional boulders. So it's quite active at times in terms of sediment mobility. So just a reminder, we're on dive 377 of ROV SuBastian. It's the deep water ROV on research vessel Falkor. That's the Smith Ocean Institute's research vessel, and we're on the Canyons, Reefs and Seamounts survey out here in the Coral Sea Marine Park. So this is actually the 7th dive we've made so far on this leg 1 of the survey, and we've completed the midwater dive component of this dive, so the descending dive, where we sampled, we imaged and sampled the plankton, and in particular the jellyfish in the water column with Dr Dhugal Lindsay from JAMSTEC in Tokyo. Dhugal joined us live via the satellite communications on Falkor. And now we're on the upward leg of the dive, observing the benthic sediments and geology, as well as the benthic organisms that Jeremy is particularly interested in. So at the moment, we're at about 506 metres depth. The water temperature is 10.7 degrees Celsius, and ship time is 12 minutes past 12 Eastern Standard Time. So we're currently on the lower margin of Malay Reef. As we go along a little bit further, we'll start hitting the much steeper northern flank of the reef, and based on the multi-beam sonar data we have, there's quite a sizable ledge and cliff face that we should have a really spectacular view of here from ROV SuBastian. So we've just put up a three-dimensional outline of the dive line. It gives you a bit of an impression of the steepness of this margin. Does it show where the ROV is, right?
I haven't got the icon on.
Right. Yeah, so we're still on the lower segment of that dive, and as we move up the profile, we'll eventually hit what looks like a cliff. And
You saw that?
based on what we saw at Herald Reef yesterday, that is quite a spectacular environment there.
Just not down. It's hard to see at that school.
Is that that rock you're looking at, Jeremy?
It was like 20 seconds ago.
Jeremy. Is that the rock?
Yeah, I'm not sure. There's a coral. I think it was in the sand. Well, just like wait a minute, let it settle down and look around.
So I saw on the monitor, that's where the RV was flying across this rock, this coral, which it's a black coral, potentially Parentopathes. So I just want to take a look. Oh yeah, this is going to be really good to collect. So this is one of those specimens that have those worm runs. So the stem usually is taken over by worms, and they make these little canals. And so I'll just
Take it offline.
yeah, please, thanks. Okay, so we're just going to zoom in here, and I'll try to show you. Well, we'll first see if there are worm runs. We're waiting for the day when one of these doesn't have a worm run, and we could find out the actual skeletal structure of this species. But once again, it has a worm run. But there's also lots of viable tissue, and there are many species in this genus, so it's definitely worth collecting. So I'm just going to zoom out a little bit to get a. Actually, I'll put it on autofocus, and then when they pick it up from the stem, we could take a close look. So can you hold it up, and we'll take pictures while you're holding it? You can get it from the very base. Yeah, it should pluck off. Oh, yeah. So you could see it has repeated branches or pinnules, and with this genus, they are in whorls. So usually on either side of the stem, there are 2 or 3 or 4 that are in a semi-spiral. And the orientation of those pinnules allow us to separate it from other species. So now that we have it, we can take a nice close-up view, and I'll just focus. Is it on manual?
Yes.
Yeah. And there we go. Okay. I'm just trying to get a really nice close-up. While it's underwater, you can't beat the image when it's underwater. The tentacles are out. It hasn't been sitting and grinding up against other corals for 5 hours before it gets to the surface. Should I move out a little bit?
Yeah, maybe come back out and then focus it there. Try to focus on that. There you go.
Okay, there we go. So, yeah, you can see. So it's very. All right, I'll spend time on the deck looking at this. So yeah, we got a good image. Let's put it in BioBox 2, please. Okay, I'll now finish my lunch. Brendan?
Okay, there's another benthic sample very skillfully acquired by our ROV pilots here, Adam and Ben. And now we're resuming our ascent up the northern margin of Malay Reef.
Sorry.
So we're now at 458 metres on our way up the northern flank of Malay Reef. Water temperature is 11.8 degrees Celsius, and as you can see, the seabed is still largely dominated by mobile sediments, with patches of what seems to be cemented areas and the occasional larger rock. And lots of bed forms in this sediment, indicating it's being mobilised by some of the bottom currents. And we're coming up to some larger boulders. So these are probably part of large submarine landslides that have moved down the side of the reef margin at some stage in the past. And I think we're starting to get into the steeper part of the margin.
Yeah, so this is us coming up on the transect starting point.
Okay, we're just about to start our 500 metre transect. Along this transect, we maintain a constant distance above the seabed with constant levels of lighting and a constant attitude towards the seabed, so that it allows us to get a consistent view and consistent measurement of features on the seabed, and also allows comparison between the dives where we've done this standardised 500 metre transect. As you can see, there's lots of rubble on this part of the seabed and a very large boulder just to our right.
So you ready to start the transect? Okay, 2 meters off. And just a little bit slower than what we did to get up here, or the same speed?
Yeah, that kind of 0.3.
0.3, okay. One other question for you.
Yeah, sure.
Just looking at where, so this is the start point, the end point is way in here. Just with the way that the ship is set up and whatnot, we probably won't be able to ascend right here. My preference would be to come back to here, come up this side, because basically what it speaks to is that the ship is down lower, so we run the risk of running our umbilical up and over the edge.
Sure. Would we not be better off just veering from this transect then just to go up?
Yeah, so I don't know if you want to do a full 500 meters or we can transect to this point.
If that end point there was to move to the, around, this whole transect is to move so that we've got safe ops, does it affect the transect? Do you know what I mean?
Right.
I'm worried if we go all the way to the sort of foot of the wall, and then have to backtrack all that. So much time we'll backtrack to that spot, but only to go here and then up.
Are you okay to transect up and over the wall?
Yeah.
So what if we go from here, and it is okay if we could, let's say we come down to here to waypoint 8, and from waypoint 8 we head due west and come up the wall this way. That way we put the ship down in here, the umbilical's nice and safe in deep water, and we're just going up the wall. So basically come down this way and then head this way.
It's what I reckon it's whatever's safest for operations and doesn't result in us having to backtrack.
Okay, so let's do this. We'll come down to waypoint 8, and from waypoint 8, then we'll get Ben. He'll show us a distance of what continues to 500, and then put a mark up here where 500 will land.
And I guess it's just an estimate of sorts between distance. What we've got is distance over the ground as opposed to distance as the crow flies for the waypoints for 500.
Yeah, like
so
this is 500 basically from here
to there.
There's that's 400.
So, but it's over. Do you know what I mean? That the transect itself is going like over.
Right.
It's the surface
distance, not.
Okay.
So.
Do you want me to measure? Do you want like a dog leg and it goes up there? Or?
Let's just get down to waypoint 8 and then play it by ear because this looks like it's quite a gnarly wall.
Yeah.
So
that's very
get down there and have a look.
Yeah, it's 100 meters higher. At least.
Yeah, at least, yeah.
And there's looks like big blocks.
That's right
sitting
here.
there, yeah.
So it should be. The nice thing about this is you'll probably see a lot of coral just because of the way the currents are coming down.
Yeah,
so
yeah.
Even hitting this corner point here might not be a bad idea.
Sure.
Okay.
Do you like really need the set length?
Well, that's what I was thinking. Maybe if I guess it's.
We can remeasure it, or you don't do a new one with a turn.
I guess we could, if we know what the distance is to waypoint 8, then just do another one that gets us hopefully up part of that wall.
Shall I give you an alternate route?
Alright.
You just keep along the route.
Onward and upward.
Thanks.
Okay, we've just worked out a slightly modified path up this steep area of the reef margin. There was a few issues with just maintaining a safe distance between ROV SuBastian and Falkor, and keeping in mind the umbilical cord that connects us, connects Falkor to SuBastian. And yeah, we're just changing a few waypoints on the fly to make sure it's a safe path. So I think you can tell we're getting close to this very steep, if not cliffed part of the reef margin by the size of some of these big reef blocks that we're coming across. So this looks like, to me, these are the deposits of a major slope failure on the margin of the reef. And these have moved down the steep flank.
Move it down.
Great, so just updating your position.
You go, Jeremy, if you want to take it.
Yeah, so if you put it even a little bit lower.
All right, this is Jeremy. There was a question, I think on Facebook a little while ago that I think I missed about white corals at this depth and if that represents bleaching. So at this depth, and then between here and deeper, the corals will be azooxanthellate, so they won't have zooxanthellae, which is the symbiotic algae that allows corals to convert sunlight into energy. So if they're white, it just means that they lack that symbiotic algae, but it doesn't mean that they're bleaching. That's just the color they are because they lack that pigment. So just because it's white doesn't mean it's bleaching. So we're doing our transect, and we're passing by what seems like another one of those black corals, similar to what we collected earlier. So there's a question on YouTube about using the corals for medical research. So directly no, but often what happens when scientists want to answer a scientific question using corals, often they won't go out on an expedition and collect corals. What they'll do is first go to museums and collect corals or sponges that will potentially provide insight into curing diseases or making medicine. So what we're doing by collecting general groups of corals and sponges does allow someone in the future to get a subsample and see if it is if it's viable for curing diseases or some other medical need. But for this trip directly, no, this is not one of the aims. But of course, every specimen we do collect does go directly into a museum. So everything that we collect, you can see if you visit the museum or if you go to their online catalog over the internet. And these specimens are going to the Queensland Museum or the Museum of Tropical Queensland, which are the Queensland Museum network consists of these two separate museums. So if you search Queensland Museum and then go to invertebrate zoology and then go to online collections or online catalog, you can search all of the specimens that are being held. And some of them have images attached. It's an ongoing process to receive a specimen, identify the specimen, upload images to the website. But for many, the images are there. No, the green light doesn't affect, this is a question on YouTube. The green lights don't affect any of the wildlife. We certainly don't see any animals shying away from the light when it's, being used to measure the distance of the critter. So we're going to try our best to just let this transect run its course without stopping. Although I've seen a few things during this transect that I've already want to kind of stop and collect, including this one right here. This is another probably parentopathias that we collected earlier. Again, there's multiple species, and because of the challenges with the worm runs that are often found with the coral, we are in desperate search for a colony that doesn't have that worm run, which will become the sample from which we describe the species and compare it to other known species. Another black coral to the right, and some soft corals below us. So these rocks are providing substrate, some sort of hard habitat, some bottom for corals to grow on. So we're seeing more of this black coral, this, we call it bottle brush morphology. So the repeated branches or pinnules go around the stem, and there's no other branches, so it looks like a bottle brush. That term's also used in botany as well, and among some other coral groups. So we are looking here at what looks like a broken black coral that is stuck up against the rock here. Looks like it's been dead for a while. Maybe it fell from the shallows and rolled down, or if it was attached to this rock and broke off. So we're going to do more rock collections and sediment samples, but not at this depth. We're going to get a little bit shallower to collect samples roughly at the depth at which would have been surface a long time ago. So we're seeing other parentopathies, and then something that looks potentially different. Hopefully we'll find another one of those a little bit later on in the transect, or after the transect's over. Seems to be a different type of bottle brush black coral. So this black coral seems to be pretty dominant at this depth in this habitat. So one of the things that we can get from this transect, since we're doing a standardized transect survey, is count how many of these black coral species are found in this strip, and that can be compared at different reefs when we do the same type of transect, to determine what potentially is driving these different assemblages. So here, it's not overly high diversity, but we see a lot of a few species. So that would be different than an area with, seeing a little of everything with overall higher biodiversity. So our questions then are, what drives high biodiversity? Or what is limiting biodiversity from being high?
About 10 meters away. I guess we just have to do this little cat like this. I think you could use that Flight's chart over there. Just basically, we're going to go whichever way the sequence first, and once we get the outfit, like that was another. But you don't have this, so okay, perfect.
as you get to the gray pixels, that's where it gets deep.
Yeah, we're gonna come due west here pretty quick and come up from there. If I come straight into the wall there, just keep them coming over that trap. I think that there's the same. See the ones that goes way out there? We're coming up here now. Let's keep in 65 seconds. Sure.
Bridge ROV.
Bridge.
Yeah, Aaron, just to let you know, we're going to go up against a wall, a big wall over here, so we're going to be doing some different, we'll just see how it goes, then we'll give you directions afterwards.
Okay, do you want me to go to the waypoint and come to a full stop there?
Yeah, just head for the waypoint.
So there's a question on YouTube. With all the samples that we're collecting, is there anything specific we're interested in hoping to get from the dive afterwards? So, yeah, the coral samples that we're collecting are going to directly help us understand the taxonomy and evolutionary history of these various coral and sponge groups. This is the first time that we have an ROV exploring the Coral Sea at this depth. And although we have collected samples from dredges and from fishing nets, those two methods usually result in the coral coming up broken, lacking viable tissue for molecular analyses. And so finally, we have this chance to collect corals, take images of them in the water to identify the habitat that they're living within and what they look like when they're in perfect condition and alive. Then we could bring them on board, and then we could take it back to where we work and extract DNA and sequence the DNA to place it in a phylogenetic tree, which will tell us how it's related to other species. And we can update what we know about each species. So what are the suite of morphological features that are consistent within this species, and how does it differ from closely related species? So that's the main purpose. Well, that's one of the main reasons why we're doing this collection. The other reason why we're collecting is because we want to understand what species live in these habitats. So the marine park in the Coral Sea in the GBR were created without much biological information. In other words, when they were deciding where to place marine reserves, they weren't really sure all of the species that lived in these habitats. So therefore, they placed marine reserves based on what we call surrogates. You know, what we think might occur at this depth or this distance from some other feature. And it actually worked pretty well. But having the specific biodiversity information, we can make much better decisions that are with more confidence will lead to better conservation impacts, like more species protected, and an understanding of what species are actually being protected. You know, you might know that a marine reserve is working because there's no fishing, but how is it working? You know, you need to understand what species are being saved because of this marine reserve, and maybe they could be taken off of certain lists, like CITES. So the number of black corals we've seen could be, well, on this dive, we've probably seen 30 or 40 of them.
Okay, Bridge, I made an adjustment.
I think we've collected maybe 15-ish black corals so far. We don't use sound to map the seafloor. Yeah, you're right. They have negative impacts on whales and dolphins and other large mammals. So we're coming up to an area of lots of rubble. Looks like rocks. Maybe we could zoom in quick. Oh wait, we're still doing the transect, aren't we? All right, we're just gonna let the. I want to zoom in and ruin the transect. You can see from here that lots of little rocks, fossil corals, and you can see there's a slope starting to get more distinct. We're currently at 286 meters, and it's almost 18 degrees Celsius, so it's getting a little warmer. Yeah, I mean, the rocks, you know, they fell from above. I guess during different, you know, when corals die and they fall, they roll down this hill. This seems to be it probably gets much steeper above this. So this might be a flat enough environment for the corals, for the rocks to settle. Jason, in YouTube, good question. Is it possible to see if there are pollutants in corals? The answer is yes. That there is research being done where they actually look for microplastics being consumed by corals, which is really interesting. So when plastic goes in the water, it'll break down, but not completely. It'll get very, very small, but it takes a very, very, very long time for it to go away, if ever. So when corals, especially filter feeding corals, when they're feeding, they very likely eat small particles of plastic. So there are researchers that are collecting corals and then looking in their gut to see if there's plastics in the corals. And the answer is yes, corals can have plastic inside of them. In addition to fish, of course, which also mistake plastic for food. Yep, we're up to this really big rock. We switch to that screen? Where is it? He's already got it. On top of it. So you can see on your screen, looking down along that rock, the sheer drop off of it.
It's the margin of the old reef.
So this seems to be the margin of the old reef. Thank you, Brendan.
Yeah, when sea level was lower, a long time ago.
So when sea level was much lower, this was the margin of the reef. So we're at 250 meters deep. Should we get a rock sample or not yet? You want to go up higher? So it's 100.
Yeah, no, that's a bit difficult to answer. All like that. Marty, do you want to?
Yeah, no, I think it's too low.
Yeah.
It's okay.
Yeah, drill.
The question about sounds, so musical, I don't know. Yeah, there are people that study sounds and the effects on different animal groups, but sorry, I don't know. I would think, and this is without knowing much about the field, that the corals eating small amounts of plastic are probably, I mean, it's not good, right? But I assume it's not going to kill the coral every time it eats a bit of plastic. But of course, as plastic becomes more common in the oceans, the more they eat it, I assume the less, the more damaging it'll be to coral communities. So we're starting to see some branching corals, which we really haven't seen much of, save for a few down in the deep. So we're seeing a black whip coral, which we haven't seen much of. So we'll resume collecting once this transect is over. So we're seeing more whip black corals now, which is pretty exciting. It's another target for this study to compare the deep whip black corals to the shallow whip black corals, and of course depths in between. And this would be one of those in-between depths.
Just let Andy know so they can log in.
We're seeing more black corals. It's an interesting branching black coral there. If we don't get too far up to finish this transect, can we come back down to here?
Sure.
I don't want to stop now.
I think it's the end right now. Want to have a look at them?
Okay, so the transect has ended. So we finished our 500-meter transect, and now we can resume collecting. So we're just going to look at this coral here. I'll just zoom in. Hi, thanks. So we're using the Rockman Sock'em to get up against the wall. Remember, the right bumper is called Rock'em, and the left one's called Sock'em. Okay. Oh, this is interesting. Right. So this looks like this is probably parentopathes as well, but it's branching. So it's it's different than the unbranched morphology. It's really beautiful. So we definitely want to sample it. It could be one of a few species. I'll just take one close view and then we'll sample it. All right, we're just getting situated here on the wall. So you can see when we get close up, there's a lot of stuff living here, besides black corals. Lots of little colonies. Oh, sure. Thanks. All right, so we're just going to take a quick look just to, so we have these laser beams, 10 centimeters, so we can approximate, well, the pinnules are pretty small, so we're probably not going to get, we could approximate. All right, it'll be easier to look at in the lab. So if we look up here, we might be able to see the pinnule pattern. Oh yeah. So you can see this is the back side of the coral, and then on the other side, the one facing away from us, has these spirals of pinnules. And of course, so the number of spirals and the number of pinnules per spiral are informative features in identifying the species. And you can see the polyps and the tentacles. Really beautiful. All right, we're gonna grab the whole thing.
Grab the whole thing?
Yeah, please. Yeah, this wall has a lot of life on it. And of course, we'll put it in box 3-4. Sure. Beautiful. Look at that. What do you want?
We're just zooming out from the camera.
What was that? Oh, it looks like we got That's okay. You can put it in the back.
Up close look at everything.
That's okay. So you can see it's got one main stem and then branches coming off of it. And then the colony is pinnulate, so on the branches and on the stem are these repeated branches called pinnules. And it looks like we got another coral with it. Maybe some sort of a hydroid, potentially. So it's a little large, so we're going to put it in the back box. We don't have our turtle with us today. Hopefully tomorrow we can be surprised with another animal, another critter. All right. So we're at 220 meters, 20 degrees Celsius, so it's getting a little warmer. So we're now going to turn our attention. We're going to try to find some hard corals as we get a little bit shallower. Cool. Look at that.
That's awesome.
Perfect. All right, we're going to detach from the wall. Okay, and we're off. Yeah, Rock'em and Sock'em does it again. So we're gonna head shallower here and try to find some cup corals, some Leptoceres, scleractinian hard corals, some Acropora, some branching scleractinian corals. Actually, can we look just at the left here?
Yeah. Sure.
Sorry, Ben, what was that?
As you were going up the wall, if you could pan it so we'll get better.
Yeah.
Yeah, that would give a really good perspective.
Yeah. So I'm just going to quickly look at this branching coral here. It's interesting. Certainly, just so it's different. So we don't see those whorls of pennules. So this might be in the family Myropathidae. Why don't we grab this one as well?
Sure. You just give me a zoom over just now.
Look up.
Just zoom right up.
Yep.
That's
So we haven't collected many from the family Myropathidae. They are a shallow water family. So Antipathidae and Myropathidae are shallow water families, which means shallow is a relative term, isn't it? When a group can be found at 8,000 meters deep, anything between 1 meter and 400 meters can be considered shallow. But of course, this is not a shallow depth. This is more of a mesophotic depth. I guess deep. But this family Myropathidae can occur in the shallows to mesophotic habitats, and so we'll collect a sample of it. Let me know when I could, I'll zoom. Well, it's hard. I guess when you grab it, we can look at it in the claw.
Yeah. I'm pulling up the starter.
Yeah, let's grab it.
You grab it?
Yeah. Oh, sorry. We're waiting on me. So yeah, we'll grab it and just, I guess, turn it so we can see the face of it. And then I think, so there seems to be a lot of interesting stuff at this depth on this rock face, so we can cut across instead of going up for a little while. Sure. See if there.
We'll do the same as the other one.
Yeah. And if you see any hard corals while we're zoomed in, let me know. Sure.
Should be inside the jaws of it.
I think you got it, yeah. Beautiful, look at that. Awesome. Great work.
Very great. It's a small chunk of habit. Very delicate. I mean, for show here, they just
Probably get it right into the box. Can you? All right, we test Ben's skills here. Can he grab it before it falls down to the deep? I think he got it. He pinned it up against the wall.
Zoom in, zoom in.
And he saves. Oh, no. If you want to grab it by one of the branches, if that'll be easy. Yeah. Ah, yeah. Nice.
You're so low like that.
So I guess the lucky thing about these branching black corals is they've got lots of, so the branches are fairly rigid, so they're probably holding on to the rock, which is saving it from falling down. And it gives Ben a chance to grab the coral, which he has now is about to have done. Great, awesome, good job, Ben. And it also has a crinoid attached to it. So it looks like it'd be in the genus Myropathias, in the family Myropathidae. Another common genus in this family is Copressopathes. So the difference is Myropathes has it's flabellate. It's not three-dimensional, so it's flat. It grows in one plane, and Copressopathes has pinnules growing around the branches, so it's more of a three-dimensionally growing colony. All right, we got it. This is a really nice specimen. Yeah. Ben, do you play baseball? All right. So there seems to be a lot of really good stuff going on at this depth on this rock face. So instead of continuing upwards, we're going to shoot across the rock and see if there's anything else that we haven't sampled yet from this depth. We're also interested in getting a whip black coral. I've seen them around. I am trying to not collect such a large one just because it's so challenging to handle and, preserve such a large coral on the ship. Like that one right there. That'd be great to collect, but it's really long and hard to put away in a box somewhere. Okay, what are we going to find next? There's a fish. I think Kai is online. Yeah, it looks like a trevally. Ooh, look at that. It's probably another Myropathes black coral, that big branching black coral. But we don't care about it. We already collected one. Oh, how about right there? How about this?
Yeah.
Okay, we found a potential candidate for a rock sample. Oh, and then while we're there, so we can get that, but then also look in this little hole. We can probably find some hard corals in the cave.
Just look up once to see what's above you. Just to make sure that when you put it, it doesn't, whatever is up there, doesn't fall on.
Right. Okay, and the goal here is to not cause an avalanche. That's the goal.
Zooming out. Zooming out a bit, just have a look.
So you're seeing a lot of whip corals. It's probably a high energy environment where they can, grow long and tall. Look at there's a fish there. Hey Kai, what are we looking at?
Some sort of cod, is it? Yeah. That's his house.
Kai identified it. Thank you. It's a cornet grouper. Yeah, Kai, I appreciate the common name.
Right there. See that one section on the right? Yeah, it's full. That might be easier to connect the forearms. See it?
So we're seeing a lot of habitat for fish to hide inside the wall. Underneath, there are corals growing. When we attach to the wall, we could take a closer look.
Just above here.
Interesting how it suddenly becomes yeah. Where are you going for? It should be, but let's try to find that. And that's me all right.
So this is a really a good example. When zoomed out, you really can't see all the life that's living on these rocks. But as we're looking closely here, things that we would have otherwise missed. I mean, if you count the number of corals right in this view zoomed in, you know, there's plenty. Different groups, different suborders. There's soft corals and black corals and hard corals. Looks like hydroids. So we're just debating where the best rock to sample.
We'll give it a shot. Yeah, go for a shot. Oh, yeah. Look at this right here.
Yeah, that's what I was going to say as well.
Okay, so you can grab that. Stretch that out. Reposition it.
Yeah, I agree. The common names are really useful when identifying different species. With the fish, there are always very charismatic common names to go along with the scientific names. Unfortunately, black corals don't really have pleasant sounding or any common names. But then again, pronouncing black coral genera and families, I think, is much easier than fish taxonomy. So I think there's just a need for common names for those fish. Yeah, this rock looks formidable. Yeah, at least for corals, we have generic names for gross morphology types. Like this is a bottle brush right in front of us, versus a planar colony or a whip coral. So, I guess we do have those common generic identifications. Certainly there's a lot of stuff living inside these rocks. Lots of worms, crustaceans, lots of invertebrates will use anything. If they can fit inside of it, they probably will. I guess the smaller the better to maximize protection against larger animals. So you can see now that we're zooming close up, what seem to be cup corals underneath the overhang there. So it might be worth collecting one of those cup corals. I don't know how challenging it would be. They're really small.
What you did with the suction sound?
Let's suction one up. You could just suction that entire area. Sure. Clean it up.
That's hard, eh?
Okay, the rock wins again.
So, Jeremy, you're talking about these cup corals, sir?
Yeah.
Okay.
Yeah. So while we're here, we are going to suction cup some cup corals. So these are azooxanthellate solitary corals, meaning they don't use sunlight for growth, and they are one polyp per animal.
We'll keep an eye out for opportunists, unless you get that little guy in a sick down. Yeah, I think it's just that's a piece of gravel for
So the angle wasn't really right for suctioning the cup corals, but you guys going for a rock sample again?
All set. Are you there right now? Yeah. You're there right now before.
And was that from?
Is that enough?
How's that? Are you we sure it came?
Did you bite that off?
Let's have a look at it.
Let's have a look.
Looks a bit rubbly.
No,
Yeah, he did get the tip, but
I think he did.
there was rubble hanging over, but underneath it. Let's just have a look. We'll be able to see what he looks.
definitely broke
Yeah,
down.
yeah, he broke it off.
Okay.
Yeah, it's definitely worth keeping.
Yeah.
Okay.
All right.
Yeah.
We got a rock sample.
But yeah, if you see another side,
Yeah.
more rocks.
Might as well fill up A, B, C, D.
We need to
Rocks.
rock and roll.
Rock and roll. Somebody asked what is in the top center of the big hole. I just see cup corals and branching antipitherians.
Are you able to grab the suction sample? Yeah, I think so.
See that rock?
I just saw it.
Okay, so now we're going to try to, if we can, reach the suction cup.
Starboard rail low.
We could try to suction the cup corals. Okay, Rockham is attached. Okay. Yeah. Once we do this collection, we can detach and look up a little bit and see what's there. Above the big hole. Yeah. No worries. So which suction number?
5 is free.
5's free?
Okay, where is it now?
It's going to be right up here on the wall. It's not a great swimming here.
So it's
Straight soft.
yeah. So it's a tough. It'll be a tough grab because he's gonna have to break them off and then suck them up. We'll give it the old college try. Oh, there we go. Yeah, you did it.
How many would you like here?
That's fine. One, yeah. That's enough. Yeah, that's good.
I'll just try one.
Thanks, Ben. All right. Well, we can now send the sample to the Smithsonian. We could send one to.
What number is that? 5.
That was 5.
What do you call it? Coral.
Cup coral. Yeah.
And then we put the other rock on 1.
Yeah, 1B. Sorry, 1B.
1B. Yeah. And then we put 2 big ones. 2 or 3?
2 and 3, yeah. Do you want to rack him or hang on?
I'm gonna rack him so you get off the wall.
Don't try to rack him here. I can quarter rail out a little bit. Just over here.
I don't think better one.
How's that?
It's just fine.
Just coming off? Yeah. Coming off? And I'm going to rack in the starboard rail.
There you go.
All right. So looking above this hole, there was a question about what that big thing was. I only see a soft coral. We can go up now.
Up?
Yeah.
Sorry for that. Unfortunately, we'll put it in. Just let me know if we turn.
Okay. So another successful sample.
Take a portrait if you want. Portrait or whatever. Portrait to say here. Portrait photo. Yeah, possibly. There you go. Nice.
Nice. All right. Should we look up the wall, see what the view is?
Sure.
All right, we're gonna try to look up for you to see.
I'll pass out a bit here too.
Yeah, thanks. Okay, so we're now looking up. It's still pretty dark up there. So this looks like to a coral. If we zoom in, it's a black coral. It's in the genus Stichopathes because it has one row of polyps. So now we're seeing lots of whip black corals, which is nice. Still looking for the perfect one to collect. So, all right, we can head shallower a little bit. Just kind of, make our way up this wall.
Can we come do like a zigzag?
Yeah, that's exactly right. Yeah, thanks. So there's going to be lots of really interesting corals hiding in this rock face here. So the goal is to find them. So we're now gonna zigzag up this wall. We still have a few hours, so for the first time on our expedition, there's no rush to get to the top. It's a wonderful feeling. So that's another, I think we saw that earlier, Myropathes. And we're still seeing a lot of this Parentopathes, which is really interesting. I mean, this entire site, from the deep to the shallows, we've been seeing this coral. And we got a sample of one of them, so that's good. So if you look out across, you could see all the little corals peeking their heads. Ooh, look at this. So this is, let's see what this is, this big branching coral. So it's an octocoral. Wait. No, it's a black coral. It's interesting.
You might want to level up, Jeremy, so it'll just fly you in. Level up your camera. So just pinch down a little bit.
Pinch down.
It'll come up. It'll come up there.
Like that.
A little bit more.
Good, yeah, just go to the wall, halfway up the wall.
Yeah, I think we should grab one big bushel of that branch. It's too big to go with the whole thing, but
Pinch down a little bit more, Jeremy, and zoom out so it can fly.
oh, sorry. So what we're looking at here is, I don't think we've collected any from the family Antipathidae. From Antipathy is the genus, so it's a black coral that it doesn't have pinnules, so it doesn't have repeated branching. So there are lots of shallow water species that look like this, but we're still 200 meters deep, so it's a bit deep to be seeing this, but it's totally possible, and we're seeing it now. So it'll be extremely interesting to first take some images, because we're not going to collect the full colony, it's just too big. So we'll take some images, and then we'll grab a few branches, and then we'll hand this off to our shallow water black coral expert, Erica Gress, who will let us know what it is. All right, so we're just gonna, so the lasers are on, so we can see roughly how long these branches are, but of course, we'll have branches in the lab. So I'm just gonna zoom in, get a nice image of, so I'll just slowly pan down so we have some information about how it branches. Can I get manual focus? Yeah. Okay, there you go. Let's go down a bit more. So this should tell us a little bit about potentially what species it is, and we'll also sequence the DNA, which will tell us a lot more about how it came to be. So you could see it has distally directed branches, so branches that go outwards straight, so they're not really curved in any way. Looks like there's branching on one side of each previous branch, which is another indication that it might be a certain species. Again, the shallow water black corals aren't really my field of expertise. All right, let's grab a handful, like, I guess like this, like here, just like grab it there and pull it up and see what comes off.
Basically, we're just, this is the wall kind of right here, and we're just going to 5 meter square like this. You shoot to rest. About 2 here going up. Is it transect now? Yeah. Awesome. What else we got going on?
Lisa, I would imagine that certain turtles are diving below where cup corals can occur. The leatherback turtle can dive to 1,000 meters, so there's definitely going to be some overlap between where the cup corals are, cleaning stations, and certain sea turtle species. Of course, the leatherback dives the deepest. Yep. Perfect. That was amazing. Thanks, Ben. So that was a perfect coral collection. This is Antipathias. 3-4 in the back.
Where you want to go with it?
In the back box, 3-4.
Okay. I'll take the camera from there.
All yours. All right, that was great. And we were able to, it was such a big colony that we really couldn't take the whole thing. So the rest of the coral it'll grow back and continue.
Three, 4.
Oh, great. Fantastic.
You guys getting praised for all the precision of your artwork online.
Alright.
Okay, so we're just zigzagging up the wall.
Okay.
But we can, at this stage, we can drive up. We can go up to 200 meters.
200.
Yeah.
Alright, go.
Thanks.
We'll let them change out real quick and then we'll start. Alright, the camera's going to be all yours. I'll center it for you.
Right. Thanks. Who would you like to tell this?
That's good.
The depth of the
All right, that was a really awesome collection.
Exactly.
Alright, it's how close the bridge is it to the wall of the shell?
Pretty much right there.
You don't want to get any closer?
I'm sure they can, but it's going to put us in a weird position.
Wouldn't like
So there's another, looks like Myropathes.
down here.
Yeah, right there.
Right here?
Yeah. It's fine.
British ROV.
Chris, you want to get your arm out there? We'll do another white balance. Sorry, they made a shame up pretty.
Yeah, I was gonna move you down to the southwest about 40 meters, if that's okay, or are you happy where you are?
I'm pretty happy here, but I can, go southwest 40 meters, no problem.
Alright, cool, man, I'll do it in just 1 second.
Alright, thanks.
We're going to do another white balance since the temperature of the water's changed. Good. Kind of looks a little unbalanced.
Righto. Yeah. That white should be white. So periodically you'll see Sebastian look at something white and then use that to white balance the screen. Now that we've come up and there's more sunlight, we have to re-white balance. That's white. That's what I'm talking about. So now you can see the algae that's starting to reveal itself.
Yeah, all good now. Let's have to be there slowly.
Thanks, sir.
All right, we're gonna head up to 200 meters.
normal. Yeah, that's a cool, right?
Oh, it's an interesting whip coral. It's coiled.
Take a closer look at it.
No, it's okay. It's too big.
It's a big, pretty one to the right.
Oh wow. Look at the basal plate on that coral. I'll just zoom in, right? So black corals have basal plates, so it's the base of the coral that attaches to the substrate. And this is an extremely large one. And actually this is another branching black coral that looks different than the one we just collected. So yeah, I think we're gonna have to grab a branch of this one as well. And then that whip coral right next to it also looks good. So we could bring, if we could position ourselves to grab one, then the other without moving.
I'm gonna zoom out real quick.
Okay.
So that one is.
Yeah, this is a really beautiful, it's so rare to see such a big basal plate. I mean, that's huge. So yeah, we'll just zoom in. Yeah, this is a good view. So we're able to see how it branches. So this one looks more complexly branched than the last one. I'll just look up a bit. Look at that, it's beautiful. So it's still directly distally directed, so the branches are still going upwards. So yeah, okay. Let's get a sample.
How much how big of a sample do you want?
Like if you can grab, like something like that, right? Or even one closer up is fine. Like.
I think I know.
Okay.
Yeah, it is. A lot. What's the difference?
Okay, so we're just gonna get a sample. And we'll have to put this one in BioBox 2. Nice. With such a large basal plate, I imagine this branch will come off before we pull the entire colony off.
It's hard to get that.
Is that box 2, Jeremy?
Yeah, box 2. Nice. Good job. Thanks. Box 2.
So look at it first, or yeah?
No, I got it. Yeah, it's all good. And then,
The one to the right?
and then, well, now I'm thinking the one to the left. We haven't collected a coiled whip black coral yet, so let's grab that one.
Okay.
Yep, so that blue plate is actually the base of the black coral. It's just really, really large. I would imagine it helps it, cement itself in strong currents. And since we're here, you see this coiled whip black coral. So this looks like...
We got auto right?
Yeah, sure.
Is that auto?
That's all photo, thanks. We're just gonna zoom, we're gonna focus. Okay. So, yeah, it looks like Stichopathes again, so it has one row of polyps. And it seems to just be facing outwards or facing us, which is cool. So we don't yet know whether it's coiled or if it's straight. We can grab it, the whole thing, yeah. We don't yet know if the way that it is, if it's curved or if it's straight, or if it has like a random somewhere in between coiled and straight morphology, whether that represents a distinct species or if it's just environmental variation, morphological plasticity. Likely, of course, the environment is going to dictate how it grows, how big it gets, how complex it can grow. So this will be a great opportunity to sequence the specimen and compare it with its spine features and determine whether what it's currently known as, whether it's the same as a species that is non-coiled or if it is indeed a different species. There's lots of described whip unbranched black coral species, but we don't know how many of them are actual distinct species and how many have just been described multiple times. That's right. Wow, beautiful.
You alright? Should I do that?
Alright, Cody and Chris.
Started slipping away.
Okay. Like, I saw this going, I might just gonna sit right here because I don't know what's happening.
That was a great grab.
Do you want to put it? 3 and 4?
Yes, please.
Right. I might be able to push it down to 2 if you'd rather put it in there.
3/4 is fine. It's the only one we have in there. It's totally fine. Okay, now let's go upwards.
Stand by. Fortunately, okay. Up with Jeremy. Yeah. Oh yeah. We look. Holy cow. Tell them about that camera right there.
Yeah. It looks like a stretched slinky. I like that.
Vertigo.
So you guys can look down the wall now. It's a long way down.
Probably 40 meter drop.
40 meter drop.
One side.
And so yeah, maybe cut it up and across.
Okay.
All right, we've done a really good job collecting black corals on this dive. So I think we're going to shift our focus now to other orders.
I'd really get a piece of that outside there. What do you think? No, not. That shadow made it look a little bit.
So you're seeing lots of these little corals growing within the crevices here.
How's that one right there?
Which one?
Little crevice right there with the rock.
Yeah, that's what happened.
So we're going for another rock sample.
I'll take a little grab in there. Hold on, let's see if I can hear that. It's pretty it's just the angle is kind of tough to get it out. I might be able to get right here and see if I can pop something off.
Flat surface, so it's not.
Yeah. I think if I can get enough angle, I'll be able to get something off of there. Right there. Right there. Right at the top of that cap.
Oh yeah.
we go.
How's that? Take it.
As good as we'll get.
Go and see, please. See.
Yeah. I'll bring it over. Let me just get stationary.
The millimeter or over?
Show me the broken face of Brooke.
Oh no. Yeah.
Joe was saying, Dan, I need to go. That camera looks great. And he changed his dress up.
Look at that rock.
Perfect. Thank you.
You like it?
Yep. Have that, it's going to break.
It is. It's kind of crumbly stuff. Okay. Where do you want it?
In C, on C.
Okay. All right.
All right, that's a great rock sample.
Come up with the camera a little bit.
That's
On that important.
oh.
How's they? Is they okay?
You are in C.
Sweet. Awesome.
Terrific. Thank you.
Thank you.
It'd be like a pebble in A. The rest of the rock.
Well, now you've destroyed all the samples.
Good job, Chris.
Now you've destroyed all the samples. We might as well just dump everything we collected and start again. All right, here we are at 200 meters deep, 20 degrees Celsius. We're now gonna drive up to about 150. And we should expect to see some changes. Oh wait, can we look at that coral?
Yep, that one right in front of us.
Yeah, the white one. Every time I think we're gonna get moving, we see something of interest. We'll never get through our dive. We'll just take a close look, a quick look, see if it's worth sampling.
That's really white. I might want to balance my thing.
Hey, Jeremy.
Yeah.
Let's get it.
Yeah.
Look good. That white balance came out perfect. Look at that.
You want to grab a piece just for white balance?
Oh no, we were able to zoom right in on white balance.
Yeah.
Looks nice now.
Oh, that's a nice black coral.
Look at that.
No. I've done.
It was successful at sampling black coral so far this expedition.
Yeah.
Focused on different.
Really, yeah. We gotta be fair to all the groups.
Big fish?
Oh, there's a fish. I don't know if Kai's on or anyone who can identify a fish.
Tiger fish.
Grouper again.
So we're just gonna follow these fish for a while, because why not? Oh yeah, we don't want to go down.
And I don't want to go that way.
Yeah. All right. Goodbye, fish.
That's pretty awesome. Yeah, it looks wicked, but it's scary.
Yeah. So that's behind you.
That's behind me.
In there, yeah.
Move the ship out.
We'll move the ship. Hold on a second. To
bring
the
him
northeast a little bit.
Yeah, bring him northeast. Okay, we're gonna have to change up our plan here.
Yeah.
Because look, see behind me? That's cool.
So there's a rock behind you
Huge.
and a rock in front of you.
How about right here? Just to see how it looks.
Cool. Look at that.
Mushrooms, cords.
Keep right here.
I'm just gonna jump and look behind me real quick.
Yeah. So we're just gonna look. We're gonna turn around because it seems like we're between a rock and a rock.
You fly into a cave?
So we're gonna have to escape this situation here.
Yeah, so I'll look back. I'm gonna come up at the delta.
Sorry, Dan, program.
How are you playing behind you?
With him, northeast. Ask me if you could change his heading at all.
Bridge ROV.
ROV bridge.
Is it possible for you to move your heading to the port?
Yeah, I'm just altering actually right now.
How much can you go for?
How much can you go to the port?
We'll see. I mean, right now we're thrusting to port. I guess maybe another 5 degrees.
Okay, every little bit's gonna help. We're in a interesting spot down here, so yeah, let me know.
I can temporarily have it more, so.
Well, wherever you end up is where we're going to want to have you stay for a little while. So just get to where you're comfortable as far forward as you can.
Okay.
Within reason, like don't go.
Yeah. All right. Cool. Nice.
I might be able to do this little tip right here.
So that's one of the unseen aspects of doing this, the dance with the ship.
Okay, this looks promising. Nothing behind, so it's active. There we go. Can go this way.
Okay.
For a little bit until he takes us this home.
All right.
Alright, come back in.
Crust.
We know our safe zone is bullshit back.
So we're seeing more of this parentopathes, branching parentopathes, whipback corals.
Goals 150. Sorry, killed it.
Everything's on.
Still pretty big open water behind.
Yeah, just don't go.
Stay on this one. Stay on this right here. Touch.
Yeah, it's like the milk is going to the light.
Yeah, so it's
Pretty good.
started from the bottom down the hill. Started from the bottom.
Okay, we're now at 170 meters, 22.5 degrees Celsius. We're starting to see more soft corals.
So good. I take him northeast, that fully toward.
It's
Different colors. Is that like a crinoid?
yeah. You know what I mean? That thing.
That thing. Go touch them and they'll freak.
on a soft coral. So we're gonna let's shoot up to 130.
130?
Yeah.
Go grab it. Remember? Every time. Oh, that thing. Don't touch it. Yeah, go grab it.
So there's some fish in view. First one to name the fish gets a prize.
Name them.
You did name it. I mean, you didn't have a whole bunch of stuff up there. Yeah.
Surprise, Jeremy. Yes. Get my water bottle.
So Kai called it a wrasse. No common name. So, all right, that means I have to pronounce it. Is that what you're saying? Odontanthius wrasse. Wassy.
That's what I was gonna say.
Beautiful. So now we just breasted the rock wall, and there's this big Myropathes black coral, but we've already collected one of those, so yeah, no, of course, always take a look. You never know what you'll see. Definitely a Myropathes. Lots of associated species living on corals like these. You get fish living inside the branches. You get crinoids living on top of them. A lot of the whip coral species, actually, the damselfish will remove the tissue on the black coral, and that'll expose the skeleton, and they'll lay their eggs within the spines of the skeleton. So if ever you're scuba diving and you see a whip black coral and you see a section of the tissue removed, if you look really close, you might see little eggs right inside the skeleton. And you'll probably see those yellow damselfish swimming around the area.
Huge.
Oh, it's not a wrasse. All right, Kai, you have to get on board so you can help us with the fish in real time.
Just to see how far that overhang goes. It's far. It's far. It's a big ledge, basically, with the undercut wall.
Okay, we're now at 150. We'll go up another 30 meters. Oh, wow. It's a sheer cliff.
Yeah. It's got that cliff at the back. What are you trying to do?
So we're going to try to show you the view here. It's pretty spectacular.
So that's no light. No light.
So this is sunlight reaching this depth, which is 150 meters. So there's enough light potentially for corals that rely on light for energy to grow that can occur at these depths. And then here's our artificial light. Okay, so we're gonna head up to 120 meters and we're gonna get another rock sample.
See where that cliff.
Olivia.
I thought you were kind of nerve wracking.
Hold on a second.
Don't want to pull a bead.
You almost did, buddy.
That looks like big soft coral.
What's that? Actually, now try to let's get your lens first.
Yeah.
For a second.
We're just going to see how far this overhang comes out before
Yeah.
I come up in higher.
You don't want a surprise?
Don't want to surprise. No, thank you.
Let's see, right? Yeah.
I wonder if I jump onto this side of the reef.
Is that?
I don't like that at all. How about that?
Okay, I'm going to hold this depth at 140 on the left side to the right. And jump over this hump right there. You want me to decide on auto the higher stuff?
Yeah.
Looks like big bushy coral up there.
Auto wires.
Popping off.
Yeah, absolutely. That's how we can get out.
Look at that. So we're seeing lots of fish and coral.
So that's where we're gonna go.
Yeah,
That's okay with you.
we're going there. We're going there.
And get a cool cinematic picture right here. Turn off the lights and get that.
Give it a try.
That.
Look at that. Oh man.
Wicked, huh?
Have you ever? You think we could settle on top?
Yeah. So we're gonna try to settle on, get a rock
Hey
sample.
Eric, I just moved you to the northeast.
Maybe up there. At 120?
Yep. You happy with your heading? Comfortable there?
Look at that beautiful view. So
Hey, Chris.
we're looking at soft corals and
Hey, go ahead, man.
probably those branching black corals.
The color of the ship, the blue doesn't really show at all, and have to go really close to the screen every time.
Color of the ship? Alright.
Awesome, thanks.
Yeah. Hey bud, what you doing?
Yeah. Yeah, please. And we're also gonna start looking for hard corals.
John,
You work for the bridge and see the color of the ship for that's
Chris, how's our umbilical behind us?
So that big bush of whip corals is probably a soft coral. To the right.
How's that rock right there?
Oh, actually, can we get that purple coral at the same time? So can you get up against the wall? I don't know.
That rock right there. Yep.
Yeah.
Perfect. That worked for me.
And
Which one?
Yeah, so a rock above that purple coral and the purple coral.
Watching us live from ROV Falkor, and this is ROV SuBastian to collect a rock sample at a depth of about 129 meters below the surface. We're on the northern margin of Malay Reef. The objective here is to see if we can collect a sample of what possibly could have been coral reef that grew here when sea level was last at its maximum low level about 20,000 years ago. Possibly there was a reef growing around the outside of the current reef. A depth of around 130 to 120 meters. So as soon as we've collected this sample, we're going to move a little bit higher up to the top of the ledge. This area we're looking at now, in front of that ledge, so we'll go up a little higher, up, looking at some of the corals in that mesophotic environment.
And it looks like there's a coral there you're about to sample.
SuBastian's using its robotic arm to try and prise off a bit of this coral limestone.
Brendan, are you there?
Yeah, so you can see the limestone here is quite hard. We've had this issue with all the samples we've taken today and on previous days at some of the other reefs. Yeah, we're going to have to find a better spot, so we'll move on. Okay, so the objective now is to try and prise off a bit of coral. Jeremy's telling me it's commonly called a bubblegum coral. The orange coral.
And I think we have online Tom Bridge. Tom, are you there?
Yeah, I'm here. Can you hear me?
So Tom Bridge is from the Museum of Tropical Queensland. He's the curator of corals. He works on scleractinian hard corals. And he's also my supervisor. And we're just trying to get him online now so he could tell us about the mesophotic hard corals that could be found in the Great Barrier Reef and the Coral Sea.
You guys hear me?
Hi Tom, we can see your video now. You're live.
Ah, excellent. Okay, cool. I didn't realise we needed to have the video on to see. Okay, a few small technical issues here. Get this to work.
We'll stay at this deck. Look up to 120. And then have a black spot to the left. I'm not sure. Can you guys on YouTube or Facebook hear Tom?
Yeah, I'm here now. Just still trying to get you guys to be able to hear me. Yeah. Trying to have keep an eye on YouTube as well, just to see exactly where we are.
There you go. Tom, how's it going?
Yeah, good. Can you hear us now?
Loud and clear.
Ah, excellent. There we go. Yeah, we're really getting up into the mesophotic now. Some really interesting octocorals and things growing here, as you'd expect. It does look like there's some scleractinia starting to appear as well, which is really interesting to see. It'd be great to See if we can collect one or two of these down at around this depth.
Glad to have you on. You can let us know what you're interested in collecting, and we'll get it for you.
Yeah, hard corals. It did look almost like it was a bit hard to see, even just when Brendan was trying to collect the rock sample before, that there was a large, or not a large, but a scleractinium down there, about 130 metres. So I'm really keen to have a look and see if we can see any of these deep water scleractinium, probably mainly from genus Leptocerus, which is a real specialist of deep water. You do find them in the shallows, often in shaded habitats and things, but they're particularly adapted to these sorts of really deep environments, which is very deep for photosynthetic corals to occur. So they're still light dependent, not like the deep sea ones. But we're really starting to be able to see them at these sort of depths. And this is about the deepest that we've ever recorded them in Australia. We did see on the recent Visioning the Coral Sea trip, some of these corals down to about 145 metres depth, which is the deepest we've seen them.
Oh, wow.
Anywhere in Australia. But, yeah, hopefully we can try and find some.
Well, what we can do...
Find some out here at these sorts of depths.
Yeah, well, why don't we stay at this 130 mark and just cut across the reef looking for some Leptocerus?
Yeah, somewhere around this sort of 130 to 145 would be pretty good if we could find them down. Just have a bit of a close look at the reef wall at around 140 metres or so. That'd be really interesting.
Okay. I think you got an email, Tom.
Yeah, I'm getting a few emails.
So we'll
remind me, sorry, it took a while to get things going. I'll have another meeting, I'm sorry, in about 15 minutes. But hopefully be able to go. It's been quite an unusual experience participating in these while you're at home. In the previous Visioning the Coral Sea trip, no scientists on board the boat at all, so we're relying totally on the Falkor crew to drive the ROV and do all the mapping and everything, and we were sitting at home in isolation and watching all these things from the Coral Sea. But it's all a bit surreal, sitting out, got someone vacuuming outside my door, and got a meeting with the boss in Brisbane via Zoom in a little while. So sort of very surreal experience to also be out virtually collecting corals in the deep Coral Sea, but very exciting. It's a really new and interesting way of doing science. I think, apart from anything else, it's actually really going to open the doors and expand how we can go about doing this sort of research, which is really exciting. So yeah, let's have a look in here at this sort of 130 to 140 metres depth and just see if we can see. Corals are probably more likely on the tops of the walls, so on the tops of the ledges. So if it is really this sort of overhangs are really going to be dominated by light corals and
yeah, as you can see here,
it'll
be bigger, so we can go across.
There's one left in the Coral Sea, Jeremy, after this.
Yeah, I think we've collected enough black corals for a dive or two. Only time to collect some hard corals. So we're cutting across at 135 meters. We'll try to get closer to the reef so we can see.
So we'll just see a lot of the corals.
Here we are.
The depth,
I think.
And,
I think we have Leptocerus here, just behind this orange coral. Is that right?
That looks like it. Let's just zoom in a bit more. So
sure. Yeah, let me zoom in for you.
Yeah, done. Very odd. Bandwidth, I might turn off my YouTube. Yeah, I think so. my screen has gone very odd. It looks like an Instagram kind of effect, sepia kind of tone, but that certainly does look like Leptocerus to me.
Yeah, let's grab this. All right, we're gonna grab it for you.
Yeah,
one of those orange.
You said that, unfortunately, I can't really see. Here's my.
Yeah.
Maybe for YouTube to catch up. Yeah, absolutely. That's clearly Leptocerus. There's a couple of them here. There's also a small one just down. Yeah, that one. There's actually two. I'm wondering whether they're different. This would be really interesting. The one that's right in front of us there. We can get a bit of a zoom in on that one.
All right, I'm going to zoom in. Let me know if it's the right one. Got it?
Yeah, that one there.
G'day, Tom. Andy Carroll here. Can you see the polyp structure there in your view, Tom?
No, I can't. I mean, I can see it's got one large central corallite.
Yeah.
So I think that's, yeah, okay, now I'm seeing it better. So this is almost certainly Leptocerus. I've got to do what people would typically call Leptocerus gradualis. Gradualis is from the Indian Ocean, so it's possible that this guy's something different, but you can sort of see that very large central corallite, so it's probably what that is. There's another coral that we've collected from the Mesophotic, a little bit shallower than this, but in places in the Pacific that is almost certainly a new genus, so I'd be really keen to try and find some of that. It looks a bit like that, but I'm pretty sure that's the Leptocerus, so if we could maybe grab that one.
We're going to grab it for you.
Great. And then there's another one just about like halfway above it as well.
Gotcha. So we're going to try to scoop it up.
They're quite fragile, as you can tell by the scientific name, fragilis, but they're usually not very strongly attached, so we should be able to almost just scoop it up.
It off. I think. You think we could suck it up, or is it better to try to pop it off with the hand and balance it back to the bio box?
Good question. I'm not quite sure. Well, I don't imagine it would be very well attached. And they're often just attached by a little stalk, but it's not very strong, as you can imagine. There's not much wave energy down there, and they don't get very big, so they don't really have any reason. But having said that, I've never dived for 100 meters, so I'm not sure. Like, I've never tried to collect them.
You haven't been to 130?
No, I haven't. So I'm not sure how well. I imagine they wouldn't be very well attached. We've collected these specimens previously via dredging and things like that. So yeah, but obviously, you know, that often damages them. So if we can,
good idea.
It maybe just to suck it up. Still having a few issues with the YouTube. Sorry, with the connection. The Marine Tech Network's bandwidth is low. So anyway, I'll watch on YouTube. I know there's a little bit of a delay there.
We'll get it for you. You want this one, then the one above it, which is a bit larger and darker orange.
Looks like there's a few on that ledge down below as well. On the top of that ledge, you can see they're really sticking out of that ledge there.
Okay.
Oh, there goes. Past the ROV.
we're attempting to use the sucker to scoop up Leptoceres.
That would be amazing if we can suck up Leptoceres. No idea.
People are asking if we've ever seen seahorses at this depth. Tom, you collected a seahorse from the deep before, haven't you?
Yes, we have indeed. Look, as most people have looked for pygmy seahorses, even in shallow depths, will know they're very hard to see. So, you know, even if you're looking right at them, you often need guides and things to really show you. So yeah, the short answer is yes, we have seen them. We were collecting a Gorgonian once, Gorgonian soft coral that happened to have in Coral Sea at Holmes Reef, which is not too far from here, and it happened to have a pygmy seahorse on it. Yeah, it's quite possible that there's actually a large variety. So a colleague of ours, Sonia Rowley, works on Gorgonians, and she tells me that she's got a large collection of pygmy seahorses that she's collected by accident. She's not just been collecting Gorgonians. So yeah, absolutely they're down here, but sampling them is very, very difficult because they're obviously very small, very well camouflaged. So trying to be able to see them in this type of thing is not easy.
Yep. Well, when we find the right sea fan, we'll try to look for a pygmy in there.
Yeah. So the Hippocampus denise, which is a little orange one of the ones that seems to be down here. That's the one we found in the Coral Sea, and that's on Anella, which is a big orange fan. I've seen a few of them in this trip, and also on Villagorgia. So tend to be orange in colour, but it's quite likely that it's quite possible, I guess I should say, that there's other species down here as well. But yeah, we haven't been able to really look for them. It's just a really difficult habitat to survey. So no one's really spent time down here looking at these things. Even people that do dive to these depths, which there is a few people in the world that do this, but even if you're down there at these sorts of depths, you might be diving for 15 minutes, 20 minutes down here, and then you've got 4 to 5 hours of decompression to get back to the surface. So even if you do make it down, you're doing very long dives. You've only got a really short period of time at these sorts of depths. So alternatively, you could potentially use some really high resolution cameras and things. But yeah, as any of you looking for pygmy seahorses will know, even places where you see them regularly and guides know where they are and which fans they're on, they can be really, really tough to see. So yeah, really, really hard to do.
Yeah, when I used to work in the Philippines, there were plenty of seahorses, and the local guides, they're so good at knowing exactly where they are and bringing guests over and pointing out the seahorse in seconds. And I would bring my group, and I'd spend 5 minutes looking at the sea fan. I know they're here. Sometimes the local eyes do a lot better than us, like the seahorses.
Where are we going here? Yeah, still having some issues with the bandwidth on the live feed, so just trying to watch it on the as much as possible on the YouTube. See even Jeremy there on even just the way we're looking at the moment. You look just to the left of the edge of that box. I can see there's a little Leptoceres down on the bottom of that ledge there. So Yeah, look, we're really keen to see, I guess, a few of these, some of these little leptoceras down at these depths. And then particularly once we get up into the shallows to see if we can find some of the more of the foliose and branching coral.
That's the plan. All right, we're just gonna try to grab it with the arms and fingers crossed.
Yeah, Jeremy, so I'm going to have to go to my other meeting, but I'll keep watching on via YouTube, and hopefully I'll send you a message if we can collect some more stuff. There's actually quite a lot of these little leptoceras down here, and it seems like there's a few different types, potentially.
Okay.
It's really interesting to see what some of these are.
All right, we'll collect a few. Thanks for stopping in.
Yeah, no worries. Thank you very much. I look forward to doing more of this as the trip progresses.
Absolutely. All right, talk to you soon.
Awesome. Thank you. Thanks a lot.
So we tried collecting with the arm and it just proved too fragile. There seems to be a lot of leptoceras in the area, so maybe we'll look a little bit deeper and find it elsewhere, where it's a little bit easier to get an arm. Oh yeah, if you can grab either the one of those two.
The Facebook chat doesn't update live for us, but the YouTube one does.
Okay.
Yeah.
It might be worth getting the shovel out, even if the black coal's not going to float away. So try to grab by the arm. You can even place the black coral on the ground there, and then separate the two.
Just try to shove the black coral to the left of it. You know what I'm saying?
Yeah.
Move to the side.
So we're just trying to figure out the best way to collect these very small, very fragile corals.
Let it fall right down to that little crevice, and then just pick up the team and
Yeah.
shake it off. You know what I'm saying?
Look at that. Kept the coral and got the shovel. All right. We're going to give this a good... We're going to give it a go.
Going nowhere?
No, it was. The bag was looking up.
Oh, it's a bit...
Vortex.
It's up on the ledge, huh?
There you go.
All right.
They ain't going nowhere.
So...
All right, now
let's get some What's the series?
It's actually perfect in there. Up here where you're holding, grab that one.
I was thinking some of this, we'll see better. Come on, dude.
I don't know why we don't like this one.
Because they're fun to make.
What is that? Is that the monkey fist?
Yeah.
It's the first time we ever put a monkey fist on that.
All right.
Alright, Curse, go for that one that's right there. Take one.
The one I can't see because I got monkeys just tape everywhere.
Yeah, go for the one. Bring it.
I think we're going to get it.
That one right there, right?
Yeah.
Okay.
Yeah, scoop deep.
Zoom in on that. I'm gonna sit. Up on your sit just a little bit.
Sit going up. Oh, you touched my thing. On top of it. There you go. Come down to the whole thing.
Yeah.
Try to get underneath it.
Oh, that's great. It looks like the whole piece underneath will come off pretty
try
easily.
to scoop more than just that.
Yeah.
There you go. You got it.
Wow, look at that.
And you go forward.
And some more stuff. Don't fall. Don't fall. Come on, gravity. Work with me. No, never mind, gravity.
Oh, what was that? That was a Gorgonian.
I can grab that again.
Nah, it's okay.
All right. Where do you want to put it?
In Biobox. Sure. A. 1A.
It's the same to you. I'm just going to keep this in my jaw until I don't need it because that was a pain.
Yeah, no, absolutely. No, let's collect a few while we're here. Even
Jeremy, do you want to click the one that was half broken?
No. There's plenty in this area, so I think we'll just
Just get it.
Yeah. So we'll drop. Tom says try to get a little deeper because this is the deepest you can find them. So, so we'll 1 in A, 1 in B, 1 in C, 1 in D, or whatever. Then we got our own series.
Well done.
Still got more stuff in there.
But I got the important part out.
That's the rest of it.
What else?
There might be some cool stuff.
Gold.
It's actually better if you don't put the rest of the stuff in there.
Yeah, what were you trying to make me do? Ruin the entire thing? Start over again?
I'm thinking about Christmas.
Then we just dump what's in there then?
I mean, really, it's getting there. So, let's drop down to the ledge below us.
Okay.
And do it again.
Hold that, Chris.
We're right here.
Foreground coming. Step that back off.
Yeah, after that. Alright, there it is.
I think like that over there, ledge. You know.
Okay.
All right, let's. Actually, I see him like here.
Yeah, now that we've put it out and seen it everywhere.
You think you could just scoop that while you're hovering?
Yeah. What do you want to scoop? One blob on that thing?
Right.
You get zoom in there?
It's like an ear.
Oh wait.
That's sponge.
No, never mind. Sorry.
That's sponge?
Yeah. Yeah. So I guess we'll keep going down to the right, I guess. Wait, is that what we just looked at?
Yeah.
Okay. I get that. Don't let me do that again.
Oh, jeez, I can still Chris.
Yeah, there'll be something on this wall.
Is this one right there?
Oh yeah, that's it. Yeah.
That might be
Ideal specimen.
Yeah.
Look at that beauty.
That one there?
Yeah.
Yeah.
Those two? Yeah, that's one. Just
Yeah. So you can grab both of them and put them in the same bio box because they're in the exact same location.
Here's my hand. It's way down. Stand by.
Pretty good. We're gonna have a busy afternoon. Okay.
How's that?
View from the shovel.
Not messing up your viz too much.
Good one, John.
There you go.
So we got lasers on the coral. We can see that they're about 6 centimeters across. Oh, sorry, Kai.
I don't know what you're talking about.
Sorry, I didn't see a fish with my 5K video. The RV is just showing off now.
Exorcist Chris. Yeah. Trying to figure out the best way to approach this.
That's not the best, but
It's definitely not the best. It would fall away from me. So
I can try to move.
Well, I'll give a shot at this one last time.
What you could do is you could drop it and pick it up from a different angle.
Or maybe you come up across. I mean, yeah, if it, there's a chance it'll pop out, but there's 2 others.
I can't see it so hard.
Got you instead.
I destroyed it.
Did you? Alright. 2 more chances.
Alright. You want me to try to park?
Yeah, park.
Somewhere different?
Yeah, 'cause the angle is coming in it.
See if I can squeeze in between these 2 rocks here. Not gonna be easy. Just trying to give you a challenge.
I failed.
It's okay though.
Good.
Let her rip, David. Chill.
Yeah, it's alright. Do this.
Okay. Attempt number something.
Got it up there to the left. Sit.
Yeah, thank you guys all for enjoying these dives. They're really fun to do on the boat. And speaking as someone who was avidly watching these live streams before coming on the boat, yeah, it's very entertaining. It's nice to have this on in the background while doing research. You know, at my university I have two screens. One's usually on this live feed and the other's on what I'm actually doing. So it's always nice to have this just playing in the background.
That's one, you're on a different ledge right there, Chris.
I was trying to get underneath it, but
that's a ledge. That's a ledge that you're under. You can see it, the outcrop. He's on top of that.
Yeah. I don't want to touch it because it's so fragile, so I'm trying to get it underneath it, but it's super hard.
Just shimmy it in.
Just shimmy? When you touch it, Nick.
Tippy tap. You're making excuses.
Tappity tap tap tap.
Tap and Rooney. There you go. There you go. See? Told you.
Here, buddy.
You get it?
Not quite yet.
Getting it. You got it, Chris.
Ooh, popped it off.
That blew up now.
All right, let's keep moving on.
You had him, huh?
I had him, but it was like as soon as you touch it, it blows up.
Let's get ready to.
All right, maybe move right. Just keep heading in that direction. If you could settle down, it might be easier. Is that right?
Yeah.
All right, let's find a place to settle down.
A really weird spot.
Yeah. Can you like sit on this? No. Ooh, that's an interesting looking coral. Nah. We're on a mission here. Can't get sidetracked by other corals. We'll try to stay at this depth and just keep going right until we see. Well, I mean, there's one, but that's hard to get, no?
Maybe.
That might be easy.
Well, if you could sit, I mean, that's probably not. If you could go, can you sock 'em, rock 'em?
Sock 'em rock. Got one. Oh, I found a perfect one. Actually, right there.
That's a fish.
Right there. See them right there?
Yeah.
Yep.
That's it. That's money.
Okay, we found another Leptoceres. We're gonna get it.
Let's see if I can scoot out some more, Chris.
Look up and stand by. 95.
Stop waiting there. Hold on, Chris. I need to rotate. I think both of these on there.
Of course. Oh. Yeah, okay, this one can be better.
Happening. Just happening.
What's that? Breaking.
They're kind of cemented in there, aren't they?
The problem is they're on there. Like the base of them is pretty solid, but the bigger part's super fragile. Take them work.
So do you need the whole thing, Andy?
Ideally. Doesn't have to be, but because they're so small. Kind of the central of the coral line. Others radiate out. But see the base there. Just wonder whether trying the one.
Yeah, they got that one on the top left.
The top left, and then got another one to you behind the monkey fist. Another. That one might.
That's saying?
Their basal it's like hard, right?
Yeah.
They're just glued to the substrate.
Side here. Just give it to you in 1,000 pieces. That's fine.
Are you truss sided? Truss sided?
Right.
And it's tone as well.
Yeah, it's
good.
Oh, no,
I got it.
Yeah, that's still great.
Did you go in?
Or pieces.
I mean, it's something's in there. It's right there.
Yeah, we'll take that.
Grab one of those other little ones next to it.
While you're there, you're kind of in the
Do
right.
you want to dump this first?
No, just grab it.
They're all
the
the
same
top
area.
one. If they're in the same location, it's fine. Alright, where'd that other one go?
Right behind it.
Maybe just make sure it's in a different box than the other one. Where's the one that was kind of sticking out a little bit more?
Right behind you.
Pointing directly at it. T-shirts. That's super cool. Yeah, that's awesome.
Ooh, 58 temperature.
Yeah. That's not fun.
I could grab this round to back off and cool down.
Yeah, okay. All good?
We can fly around and look at stuff. I can't land out.
Yeah, it's fine.
So Jeremy, I think the margin of these colonies, particularly when these tiny little ones,
Yeah, that's fine. Should we just leave it? Let it be?
I think we're in the right spot. It's really going.
Okay.
But that's why it's already as you go out, so it's always get in there.
They're essentially, let's run across this up.
Yeah.
All right, all good. So we'll put it in a different. Is 1D?
When D has the bits in it, has something in it, like rock, that thing.
How about 1B? What's in there?
1B? I don't know, just a circle. I don't know what it was.
1B has a rock in it.
Let's put it in 1B. And then, yeah, we can get off the ground and zoom around a bit. But we don't want to go shallower yet, right?
60 Celsius. Is that bad?
Do we want to go shallower?
No, not at the moment.
Okay. At least not for a while.
What's that?
Not for a while, or we kind of hit our maximum?
No. We can definitely go shallower. Oh, 100%. Okay. I just need to cool down before we go shallower because I'm already at 60.
Yeah.
And my sub was about to peg out.
Okay.
It's because we're using both arms, Dad and Trey.
You're running hot.
I should get that in, Chris. I had their own view.
Yeah.
Yeah, a pair of tweezers would be fantastic. Not enough arms on this bad boy.
Something fell out, but I don't know what it was. It fell in the...
I guess it was the coral. Ah, there's.
Good?
Yeah.
Alright, well done.
Close first.
Alright. Let's never get those again.
Except for that's the next we're gonna get. But yeah, we can zoom around a bit. Maybe keep going right.
Chris, you're fully locked up.
Yeah. Well, as soon as
take that over for a break.
Alright, I'll still it real fast.
Yeah, go still. Come back real quick, get some water flowing through my hydraulics.
Alright, buddy.
So you come down a little bit, get a little bit. Look like a come to the right.
Not a 59.2. But you can't get to 54 in 3 seconds. 3 minutes.
On the LiDAR, we're seeing a couple of terraces still above us. And each one of those will be a sea level wave terrace, so we're going to want to do lots from
Rocks.
Yep, lots of corals, a lot of fan corals here. We're looking at soft corals and black corals. And fish. So we're not heading to the surface just yet. We've got time, we've got all day. So today's an ROV day, and then on the second leg of this trip, which is coming up pretty soon, we're gonna have ROV days almost every single day. So you're gonna get bored of the coral sea exploration. So the reason why we finished collecting that Leptocereus, and we just want the ROV to cool down a bit. So we're kind of hovering in mid-water, letting water run through the system to cool it down, and then we'll go a bit shallower and sample some of the shallow water biodiversity.
Would you like me to come up as a this, Jeremy?
Sorry?
Would you like me to come up as I do this?
Yeah. Yeah, of course, that was sarcasm. Who could ever get bored of this? And so we're in the Coral Sea now, but we'll be moving towards the Great Barrier Reef, the canyons. So, you know, it'll be a slightly different environment. We'll see different things, different latitude.
Sorry, do you want to chair back a bit further in the lower?
Oh yeah, look at that.
That's a nice rock.
That's cool.
Yeah. Tom, how many species of Leptocereus do we have here? That would be suitable for popping off, or?
Yeah. I can forge out too, so I can use less power.
There you go. So we're gonna yet again collect the Leptocereus. There's just so many, and typically they're hard coral to collect because they aren't found in the shallows, usually at the deepest depths of recreational scuba diving. So you can find them at about 40 meters if you're in the right spot at the right time. So being able to, you know, casually be at 150 meters is an opportunity to collect these Leptocereus corals.
Yeah, sure. Go get it.
That's kind of what it is at the moment. Should we grip, do you think?
Here, I get something cracker. It's barely on it.
Yeah, now it's inside.
Yeah, you should probably grab that. Yeah, it's trouble and press trouble.
What did I do now?
Stay with you. See? Told you. Shoulder came in, hands, arms across.
Okay, I saw you.
Indiana Jones hat. Back up, Chris.
I'm trying to figure out how I should grab it now.
You better hurry. Get not fast.
Great. Alright.
Same way.
Same way, just a better grip. Yeah. So.
All right. Feel good about this one.
A lot of pressure.
Seems pretty hard under there, huh?
Trying to get anything below it. So
Yeah.
like,
Oh, all right, that's all right.
hold on. Is it, if it's in pieces, is that okay? Or you need to
If it's in 2 pieces, it's probably fine. Yeah, if you can get that full piece, that'll be all right.
I will try.
It's tough, huh? Oh, now you ruined it.
No.
That's all right. It's too close to the rock. It's
I got a little bit.
toss it. I'll be right. I feel like if I say toss it, Tom's gonna get real mad that I said toss it.
Put it in. It's a pretty decent sized piece.
Put it in bucks too. Nothing. There's no other lictosers in there, and you could put the whole shovel in there as well.
You over this? You done?
I think so.
That, I think so, was almost as bad as I'm not mad, I'm just disappointed.
We may not be done with the scoop though.
What's that?
We may not be done with the scoop.
You think we'll use it again? I can set it in there better now.
Yeah, just put it on top, I guess, so it's easy to grab.
So let me put down with this. Here's I have my need one without it.
Great, Crystal. Get warm.
It's feeling great.
Coming up to 58.
Beautiful. Now the goal is to not drop the shovel on the coral.
I was going to set it in there.
Oh, you're putting it in?
Yeah, put it in.
Go ahead.
Yeah, that looks good.
Great.
Nice.
Cool yourself down, buddy. Go forward, so there you go. Now shoot it.
The bulb is not in the vessel, so it's still stream, but it's very good. 6.5 and of the walls, very steep.
Alright, let's get some rocks.
Yeah, rocks.
Rocks on this side.
It's more of a fish guy, so
I'm not collecting any fish. I'm trying to grab right there in that center.
I'll see kind of before you start whaling at o'clock. But yeah, it looks super great. Where we are.
A fish.
I don't know, I'm just zigzagging right here. Yeah.
Control deck.
You go ahead there, Deck.
Hey, are you guys coming up a little bit more? We can put the pinch wheel on if you're coming up shallower.
We're steadily going to be coming up, but right now we're just kind of moving around trying to cool the temperature down a bit.
Okay, what's your depth at?
135.
Okay, cool. We'll just keep looking for the floats.
Roger. Is the umbilical drooping at all?
Not too bad. It's leading upward to your our starboard, ship starboard.
Okay.
But we probably could turn on the pitch wheel as long as you're only paying in.
Can't guarantee that.
What I'll do is I'll engage it, and if you want to pay out, just let me know.
Alright, deal.
Cool.
What's next target depth?
100?
Can we throw off the water? See these terraces?
Terraces.
Above. That's what we want rocks. Okay.
That's the.
Yeah.
Oh, this was.
Yeah, I was like, she's dead.
We're watching ROV SuBastian. We're in the Coral Sea Marine Park, Malay Reef. This is being broadcast live for the internet from research vessel Falkor, Schmidt Ocean Institute's research vessel, on the 7th ROV dive, Seamounts, Canyons and Reefs research cruise through the Coral Sea. So we've just finished, a little while ago anyway, finished our mid-water dive where we ended through the water column down to about 630 metres, and during that dive observed and sampled plankton through the water column, in particular jellyfish. And once we reached the bottom, we started our ascent up the very steep margin, southern side of Malay Reef. We've come up some very dramatic ledges and cliff lines that seem to delineate the old reef that formed here probably millions of years ago. Now we've come up into the shallower reaches of the reef margin. We're in water depth of about 126 metres. The objective here is to collect specific coral species and also to attempt to collect some rock samples from the reef limestone. The idea there being to try and find rock samples of the old coral reefs that used to grow here during lower sea levels. A series of terraces that we've mapped. We've mapped using the multi-beam data. About 3 to 4 terraces, shallower part of the reef margin. The idea being that if we can obtain some samples that may be suitable for dating, then identification of composed of reef material gives a bit of an insight into how these reef systems have evolved over time. Really recording past lower sea levels or periods of lower rising or relatively. Okay, we're on top of one of the ledges that we have been able to map in the multi-beam data. Very rubbly surface on the top. Some beautiful looking fish here. And here we're looking at some sort of large hole, probably formed by fish, we think. and lots of rhodoliths. These are roughly spherical concretions formed by encrusting red algae. So we're just positioning ROV SuBastian to collect a sample of these rhodoliths.
You gonna use the board arm? Nope. Nice little job. How about you grabbing that? Back? Yeah. Grab that back.
So you're watching the robotic arm of ROV SuBastian rather, and this scoop should be really effective at picking up these rhodoliths. Oh, look at that.
Unfortunately, it's spun. It's spun in the wrist.
You've got two hands.
That sounds nice. That sounds.
Keep shifting. Shift it. Keep going. There you go. Shake it. Do it. Yeah. Fill that thing up.
Mm-hmm.
Christmas present for everybody. Everybody gets it.
Looks like we got a sea cucumber. We haven't seen many of them on these dives, but here's one.
It's like he's asleep. Keep going up.
Yeah. Yeah. So we're going to keep exploring. We're now at a little over 100 meters deep, and we've still got time on this dive. That's a soft coral. A lot of rotoliths. Mounds of rotoliths. More soft tree coral. A lot of soft coral.
I'm probably in the ship right now. Once up this lights up.
Oh, I'll move down south, west.
Yeah.
By here.
That works.
Bridge or ROV?
ROV bridge.
Hello, Eric. I moved you to southwest. You okay with that?
Yeah.
Wonderful, wonderful. Thank you. Alright, I'm gonna bring in about 10 meters.
Yeah, copy that, 10 meters.
Yeah.
Oh, that's interesting.
Is that right?
It's a weird looking soft coral. No, it's okay. It's a bit odd, but. Okay.
Take a nice picture of it.
Let's take a nice picture. So you'll often find lots of brittle stars and little crustaceans on these soft corals.
Difficult, Chris.
It's like we're flying over little buildings. Another sea cucumber. Looks like a flat field. But we are going up. We're now at 100 meters. It's 24.3 degrees Celsius. It's anemone.
Another fish hole. And all the one.
What's the shallow part here, John? The shallowest? Just so I know if there's any kind of
16, 17, 17.
No, minus 70 meters.
70. So nothing too big, extravagant to get from boat to call.
From where you're at right now.
Pretty flat.
It's pretty interesting plateau.
Deck ROV can pull another 10, 50 meters.
Copy that, 10 meters.
Maybe 15.
So we're now at 90 meters and rising.
Just working my way this way. So if I just look this cliff right here,
We'll hit the tippy top.
Control deck.
Go ahead there, deck.
Yeah, just so you know, you're leaning hard to vessel starboard.
Roger that. We're bringing the ship down so it should straighten out. But keep it down. Cody's running pretty hard. Yeah.
Cool.
Is it ready to zoom in?
Yeah.
Yeah. So while we're exploring and heading towards the shallows, we're still looking for scleractinian corals, not leptoceres. Smaller branching hard corals.
Yeah, how comfortable are you going south and southwest?
They're often so small that they're
Sorry, say again?
How comfortable are you
overlooked.
going to the south and southwest? Are you nervous about that at all? Or
But if we look closely, we could see what's living
Yeah, it's fine right now. I mean,
down here.
It's
So
go any faster than that.
No, not the speed, just how far. I don't know where we are as far as the depth of it.
So I'm just gonna look around a bit.
Fine. But yeah, Jason's gonna take over, so.
Alright, I adjusted you a little bit south as well.
Alright.
Thank you.
So these are bryozoans, these little netted corals. The thing in focus now. So yeah, those are those are made of calcium carbonate. Those are hard, brittle bryozoans. I've collected those before. Andy, what do you think's in front of us here?
See that coral there at the base, Jared?
Yeah. What do you think this is?
Yeah. Hard square.
Yeah.
Should we click on easy do whatever? Yeah, the whole thing. Yeah, I did this. You have to rip it up.
Arm wrestling. One more.
I'll do arms.
We're gonna grab this. So this looks like a hard coral.
Oh, your motor temperature's pretty hot. It's at 57. I'm sitting down doing a quick sample coming up, slowly flying, not that much thrust. I have a little bit down, a little bit forward of the position. Ship swimming down here.
So you could see in focus now the coralites.
Yeah.
So we're gonna click this and see what it is.
If you look at the 3D render map that John has, this is damn near all flat. Okay. So we're just cruising around.
We're gonna grab this coral.
Okay. Crew change here.
Yeah, so we're looking at this coral here, this branching coral, and then there's also a coral underneath it, which we'll be able to get a good look at once we remove this coral. So this is the beautiful thing about just stopping anywhere. Everywhere you stop, there'll be something to see. Ooh. We have a shark. We're gonna try to give a view for everyone to see. We'll come back to this coral.
See the waypoint there?
Cool. Did we miss it?
It's behind us right now.
It's behind us. Behind us. All right. While we collect this, we'll wait for the shark to come back into our view. Let me know if you need help. Vision-wise.
We're gonna click this guy.
Yeah.
Where would you like it to go?
Oh, I don't know. I guess 3, 4.
3, 4.
Yeah, there should be room in there. Got a lot of stuff in there, but
Getting it from as far down as possible.
Just gonna zoom.
yeah,
Just I didn't quite see where this was.
sure. Yeah, the next time we see, oh, there's the shark behind us now. Is that a shark or is that a fish?
That one's a fish, but there is a shark up top there as well.
All right, so there are sharks in the area, so we'll try to get you a view of the shark.
Just that train.
All right, we got J-Rod trying to collect this coral. That's a beautiful coral. I think we're gonna get the cup with it. Yeah, it's really good. Yeah, you can. Get a rock sample while we're at it. Unless it's huge.
Unless you don't know what you're on.
If you take that whole thing, you can put it in 1D.
It looks like it's too big for 1D, the rock. Is it? I'm not sure.
We'll find out here in a second.
Cool. Where do you see what Rob said?
Sorry.
Do you see what Rob's comment is? What type of Alameda is this?
No species.
Huh?
Like no species.
Okay.
You know, there's two there, but
There seems to be two types of Alameda that we're looking at here. Sorry, Rob, we don't know the species. Thanks, Luke. You're on top of it. He said there's at least two species.
It floats on surface.
That makes sense. The ship should be coming due south of us here, so should be starting to straighten out.
Roger. It's pretty straight now.
Okay, thanks.
So we're at 87 meters and 24.5 degrees Celsius. And it's a really interesting habitat here. Lots of halimeda, and obviously corals that are really hiding within the halimeda fields here. And so one of the target species is this branching acropora that hides within halimeda fields. I'm not sure if this is the exact one, but we'll see. And then we're also going to try to find some acropora, if possible. Yep, this two birds with one stone here. We get a rock sample and a coral sample.
Well, I was, but now there's two. I just want that guy, right?
Now that it's moving, it seems to be a soft coral.
Still want it, right? Yeah. Still want it, right?
Yeah. Jeremy, they're talking to you. This one you still want it, even though it's a soft coral.
Sorry. Oh, beautiful.
Am I putting this in again?
I don't think it's going to fit in 1B. One of the bigger boxes is probably the best,
It might fit in 1B.
You right?
Maybe.
D, if it fits in a small one.
Suppose. Okay. You're going to try 1B first? Right? I think
What 1B's okay for you?
Yep, sure. We won't confuse that with anything else in there. Yep, so we got a soft coral, some halimeda, and a rock, and I'm sure when we look at it on deck, we'll find more stuff on this rock. Every rock sample we bring up, we find more than just rock, so yeah, this is a good sample. Very nice. All right, we are filling up these bio boxes.
So while we're here, I'm pretty keen on a sediment scoop. We need to think about where we're going to put it and how we're going to do it. So not necessarily a scoop of the soft stuff. And there's a whole bunch of forums there that we're interested in. And
I can also do the suction.
I don't
They might clog up, so it goes good.
I'll get the point. Can I get the pointer and show you the forums that we want?
Hey, you just pick them up.
But no, we're going next week, so we get Alameda and we'll get as far as the ones in the a whole bunch of stuff.
She wants to keep.
All right, while we're here, we're going to get a sediment sample. Good thing you got that monkey fist there.
What about it?
It's good to grab. On the Investigator, one of the guys gave a course on how to tie the monkey fist to everyone on the ship.
Did you get good at it?
I couldn't do it. I gave up and threw my monkey fist at the wall. Yeah, the fun thing about these samples, especially ones where you kind of just settle down anywhere and look around and see what you see, is anything can be an undescribed species. Of course, the smaller the specimen is, the more likely it is to be undescribed because it's just harder to see and therefore less likely to have been sampled before. So once this, we got a little soft coral there as well. I'm not sure if we'll make it in the scoop. Oh, yeah, it's in the scoop. There we go. So after the dive ends, our work begins. We have all these samples to process, take subsamples for tissue, for DNA work, and write down all the other information, the lat, long, depth, collate images from the ROV and take our own images in the lab. And then we do it all over again tomorrow. So on YouTube there's a question about keeping the samples for a while to observe before preserving them. Generally, no. There's a black coral, a little branching black coral there. It's important to preserve them as soon as possible. A lot of the species, especially ones that have fleshy tissue.
We don't need more from here. That's okay.
Where do you want it? Sorry, which file box?
Is there you make the decision, I guess. Where's the best place to put it?
So I'm fine if you dump it into D, 1D, if you can.
1D, sure.
D.
So we preserve the samples as soon as possible. So a lot of the specimens will start to lose their tissue really quickly. And Rob Beeman identified one of the benthic forams in the sediment.
Yes.
Yeah. Thank you, Rob. Rob is eager to get on the ship for Leg 2, where he could join us in the wet lab and help us identify all the different things that we're collecting. And I'm sure he's keen to get his hands wet with ethanol.
Yes, good question.
Wonderful, thank you very much.
Is that all you want, or do you want some more?
No, that's what I'm looking at.
All right, so we're just discussing the next plan. We're going to head to the shallows. We're going to see what kind of corals are hiding up there. And we're getting near the end of the dive because we are running out of space in our bio boxes. And although we've recorded what corals went where, there comes a time when there's just too many corals in a box to remember which one was collected when. So we have one more target in mind. So we're going to try to get an Acropora hard coral and simply video the top of the reef.
Maybe a line. Just saying that
Ben, you got it?
It's very quite slow.
Roger.
How close to the float?
It's about 30 meters away.
Okay, good. I can come in just a little bit more?
Yeah.
Forward here.
You start going away from the vessel.
Just keep picking it up a little because I think.
I want to give us room to be able to come in tomorrow.
Let's hear a big whipped black coral. See some fish. Well, look at that, a sea fan. Big, thick black coral.
Because we've turned into it, we couldn't see on the back.
Yeah, we're looking forward for Rob to get on board so he could join the communication on this side. Yeah. Yes, we will collect the sea fan like that on another day. Oh, that's an eel, a ray. Look at the ray there. So we got some sort of a ray. There's a lot of species. Yeah. Look at that guy. Nice. I assume once he hears us, he's gonna shoot off. Beautiful. Look at that. Oops. Look at that guy. Beautiful. All right, so we're starting to see plating hard corals here. So we're now going to look for an Acropora. So as we go a little bit shallower, we'll probably come across it.
So you're looking for an octocoral?
No, a hard coral. A table, branching. You were close.
Just floating up. I didn't want to scare
Sure.
Steve.
Yeah. Stevie was happy with us. So we're seeing lots of plate corals, soft corals, sponges. That's a big sponge to the left. Yeah, now we're just looking for a branching hard coral.
Do you want to just embed him up behind you? Or move him to port and I'll follow him to port.
All float submerged.
How about over here?
Yeah.
Bring a jar of the Bring a jar of
So Jeremy, Tom was keen to sample one to one or two to sample one.
Yeah.
Yeah.
All right. Yeah, let's grab when we can.
The white brown margin. Yep.
This one?
the plate.
Yeah.
Whole thing?
No, a fragment. To the left?
Top left. Oh, sorry, left a little bit. Oh, sorry, which one? I thought you were looking at the big one there. Just point at which one?
Andy's saying that one up in the
Oh, I'm sorry. Okay.
All right.
Pointer.
Thank you.
That one?
Sure. I don't know. This is where it gets a bit difficult. The one with the
That's
white
right.
rim?
Yeah, why not?
I mean, I should try and touch base with Tom to find out specifically what he's after here.
Just right up to the side of the building. Yeah.
But yeah, I guess.
Yeah, let's just settle down here for a second.
There's so much going on here. That's probably different. That's different. What we were just looking at was different.
Yeah.
Might be able to pop off a nice sample.
See, the challenge here is there's in each bio box there's some hard corals now. So I think we should just collect one.
Yeah.
And then another dive will, do more. Yeah. All right.
What's the plate, Jeremy?
Yeah, we'll grab a piece of this one.
A piece of the white rim?
Yeah, but so we'll try to get like a deep slice.
Nice slice of the beach pot?
Yeah.
I watched you do this the other day and they're pretty brittle.
Yeah. You can just drop the hammer down and then a big piece will break off.
I was going to try to go underneath it.
Yeah. Right, and then balance whatever comes up. That's a good idea.
Where are you going to want it to go?
Oh, I don't know.
Want to go in the back?
Yeah, 3/4. Oh wait, I think it's already loose.
Can it go on 3/4?
Yeah. Yeah, you're gonna have to break a piece off. The whole thing will come up. Actually,
You don't want the whole thing for sure?
no, it's too big. You could even, yeah, I guess at this stage just grab a bit and see what happens when you pull out, bend it.
And you want to put this in 3/4?
Yeah.
I can always just break it on top of 3/4.
If you just twist it now, you'll probably. Got a sponge underneath. Sponges. Yeah, if you break it like a potato chip.
No, just thought we might as well look at it.
Oh, no. Yeah, good idea. Nice. Perfect. That's exactly what we needed.
Would you like this to go?
3/4, please. I guess we could zoom in for a second. All right, that's a Montepora. Beautiful. All right. And then we have one more coral to collect, and then we'll be done for today. Thanks, J-Rod.
Thanks. Try for Cropra, Jared?
Yeah.
Which one we going for?
A copra? A cropra? Aquapora.
Is it right here or somewhere?
Nah, we're going to have to look for it. So it'll be a branching hard coral. So if we head shallow, we should come across one. So we're going to look for one more coral, and then we're going to call it a day. It's been a really successful dive. We took sediment samples, rock samples, coral samples. We explored the deep and the shallow. Yeah, we're going to keep looking, but I'll thank you guys for watching along, for commenting, asking questions. There's lots of plate corals here. This could probably be its own dive. We could probably just sit down here and just collect all these different plate corals, and it could be someone's research project for the next 2 years. See an angelfish in the background.
Yeah. Yeah, we'll just keep that.
Great.
Just ask them if it's clear comes or at work.
Probably turn them on. Everything's just jammed. It's so constant. It's constant. It's what we see.
So you could see how diverse this area is. I couldn't tell you how many hard corals species there are. You can see how different they are. How many different genera we're looking at here. And of course the soft corals are, and then the fish, tons of fish, lots of color.
The dark branching. Just up to the right.
Question on YouTube: do universities? Well, does James Cook University have like a mini submarine? We do not, but that would be fantastic. This is the next best thing, though. Oh, wait. How about this branching coral here? This might be a soft coral. I guess we could check quick. Nah. Oh, we'll keep going.
Yeah.
Yep. See some mycelium. We're seeing mycelium, Pachyceres, Montipora, a lot of mushroom, coral.
What's that toa?
That thing to the left? It's a sponge. Maybe it's like elephant ear, I think it's called.
It's like, oh.
Yeah. If you waft it, it'll move. Yeah, some kind of sponge, I'm not sure. Phase sponge, elephant ear, maybe. Okay, we're still looking for Aquapora.
That fish.
Amelia, that's a good question. How does one do these kinds of surveys? Does it depend on what you choose to specialize in? Or do you get a lot of generalists on the surveys? Usually, someone will propose a research question to a ship like ROV Falkor, and if they accept you, then you have a main target, and you bring along researchers that help to answer that question. And for this example, for this trip, we're trying to understand the formation of these habitats and how they enable biodiversity to live on these habitats. So we have geologists and we have biologists, and then there's also opportunities for others to join, where it's no extra effort to bring another person on to answer a question somewhat related to the primary question. So for example, my PhD is on black coral taxonomy, and we're collecting black corals on this expedition, so it allows me to collect for my PhD and also assist the expedition by collecting and identifying all the black corals that we find.
Actually, I think it's better, especially on the 25 is much better.
Okay, we're still looking for an Acropora.
Hey, Jeremy, probably we'll be lucky to find proper 95. It's normally the shallower. It's the it'll be it's more that deeper extent for a lot of purposes. So we try to shoot up a bit higher. I think if we want to, sample it.
So we could maybe go a little bit faster to get to 70, 60.
I think we're actually getting deeper here. Yeah.
Okay.
I'll turn back here. The only issue is we need to have a look at our umbilical.
Yeah. It looks pretty stretched.
Yeah. We just don't want to. We're so close to the reef.
Why don't you just disconnect? It's about time.
Sure.
Cut that cord.
Really good at his duties.
Roger that. Which way are you going to go at?
Well,
it's going to be much shallower here.
Liam, your question, does the researcher proposing the research also invite crew, or does Falkor have their research team work with them? Usually, the team proposing the research will bring along their own researchers. How did Dhugal become a part of this dive? That's a good question.
I think Dhugal collaborates with people involved.
Dhugal was in it from the start.
Yeah. So Dhugal's one of the main researchers that proposed this research project. So he's been in it from the beginning.
Because it's looking at the mid-water plankton associated with these reef
Yeah.
structures.
So yeah, it's another piece of the puzzle, right? So we're understanding how the reef was made, but there's also interactions between the reef and the plankton that falls down to the reef. So it's great to have researchers focusing on different things, but essentially we're all trying to answer the same question. The who, what, where, when, and why.
Yeah, sure. You could just push and then usually, Brendan, you happy just to map in the rest of the holes we have around this area and kind of expand up from there. Absolutely. And then likely a CTE here again in the morning.
All right, so we have about a half hour left on this dive. So we're just going to try to get to the top of this reef, see what's up there.
Yeah, we went a little bit. We're starting to roll back up. Yeah. Right.
It's amazing how many corals down at this depth. You know, when you scuba dive, you think of the top of the reef, you know, a crest before a wall to be the highest diversity. And then usually, you know, you see a drop off, you see some branching corals on the wall, and then kind of drops to the black, and you don't really see, you know, all of this. This is just 90 meters deep, but the diversity is probably higher here than it is at 30 meters. So Tom Bridge is calling this a leptoceres field. So all those brown plate corals are leptoceres. So once again, we could probably just stop here and fill up our bio box with samples. We won't, but we could.
Just going back down.
Hey boys, looks like you got 5 photons for us.
Roger that. We're still slowly working our way up, but we're going to stretch ourselves out at the same time.
Sounds good to me.
Roger that.
Is that a big stingray that y'all saw over there?
You mean Stevie Ray?
Little Stevie Ray.
So Liam on YouTube has a question about if there's a sudden change in photosynthetic coral assemblages to filter feeder assemblages, or is it more gradual? Yeah, so there's a distinct change, and this is one of the main questions. Well, this is a important question that we would try to answer is, you know, at what depth, at what temperature, at what amount of sunlight is there a distinct shift in these assemblages? I mean, they said the shift is clear. You know, as we were coming up this wall, there was a point at which there was, you know, none of these corals, and then all of a sudden it was a field of these corals. So it's a very distinct change, and that assembly change is what we're trying to understand. There are a lot of drivers at play, though, so detangling those multiple drivers is important, and so that's why we're doing things like these transects, these 500-meter transects at different sites, because of course in one location what allows for high diversity or a certain assemblage to occur will be different at different sites. Is it right to zoom in for a bit? And actually, can we slow down here? Sure. Maybe. Yeah, we'll just keep looking at that. We might collect that coral there. Why don't we go ahead and grab this? So one more leptoseries.
See where you're looking at. There's that one right there. Yeah, just if you zoom out there, just see. Ah, it's to the left.
So.
Grabbing the whole thing. Start with number one.
A piece of the pie will do.
The branching coral, right?
No, the plate.
Spanish dancer.
Oh, Spanish dancer.
Spanish dancer.
Look at that. So this is the first thing of our trip, and they're found in the shallows. This is a Spanish dancer. It's a type of nudibranch. So we're gonna collect this leptoseries. Although we thought we were done collecting leptoseries, this seems to be a different species than what we collected in the deep, and we were asked to get a sample.
The one just right below us, right?
The one here. Yeah, right below us. So this could be Leptocereus incrustans. It's certainly a different species than we were collecting when we were down below 100 meters. So we're going to get a piece of the pie here and put it in BioBox.
Go ahead, Adam.
Hey, Ben, pick up 24 meters?
Roger that.
Hold that. Stand by. I got it.
Okay.
Just let me know that's close to low, sir. Close.
Roger.
Well, I'm gonna mix up if there's this. Are you sure there's no hard corals in there?
Okay. I mean, it'll look different. Yeah. So we'll put it in BioBox too. Oh, can you grab the whole thing? Sorry.
Are we looking back?
Like the whole branching thing on it?
Got
as
room
well?
to wiggle.
Okay.
Try to,
And I'll stop there.
Roger.
What's that? The whole branching thing?
if not branching, but this whole thing. I don't know. Yeah, we'll give it a try. Try to get as much as you can. Or whatever's underneath it. I mean, I don't know how much else is attached to it. Oh, it may pop off. Nice. Yeah, if you just break this, just like crush it. Yeah, awesome.
So you said buy box 2?
It's a bit bigger now. We can put it in 3-4.
Okay.
Gross ending, gross.
What?
Instead of salivating over this. Yeah, this is a paradise for anyone working on hard corals.
Maybe just 3 of them?
Oh, yeah, that one.
All right, pretty good, pretty good. Nice.
Very well done, thank you.
All right. Now we continue.
You want to have a closer look at that Spanish dancer, or?
Yeah, let's take a look.
Sure. Why not?
We don't touch wildlife.
They are very fragile.
To the left?
Yeah, I think it was a bit to the left. Yeah, there.
Stuff like that skin is really
So I think we're gonna try to take a look at this Spanish dancer. This is a huge nudie.
it's massive, but I thought it was a turtle.
Yeah, as soon as you go forward, don't you?
Anyone, any ideas of what to name this nudie? I think the name should be Spanish.
Something Spanish.
So zoom in a little bit, Jeremy?
Yeah, that'd be great, bro.
Yep, I gotcha.
Wow,
look at that.
Find out how big, because it's.
Oh yeah,
it's
lasers
quite-
would be good. It's huge.
You want lasers, you say?
Yeah.
Yeah, just because it looks quite bigger than.
It looks fake.
The lasers right there?
Oh, it's huge.
Yeah, that's not.
I can't turn right anymore. Oh,
Brad, just slide in.
that's right. I'll get center. So this Spanish dancer is like 40 centimeters. It's over, maybe it's 50 centimeters in length. Beautiful. So you can see its gills hanging off the back. I'll just try to zoom in a little bit more.
Yeah, I can take that nice big fish somewhere else. And be like, have you seen the penis fencing?
Looks like he's hiding his head.
So when he brings both males, and when they mate, they literally try and kiss.
Consuela. All right, we have Consuela.
One is successful.
Not a bad name.
Pass me point to the penis or wherever.
Let's see if there's anything here we can look at.
And they do these beautiful rosette.
Yes, I've seen those.
Oh, so you see shrimp on the gills section.
Eating the poop. It's like it's still during the poop. It's like a hot rod. Can you see the shrimp in behind it? Translucent shrimp. It's behind the gills. It's filtering its poop. It really
Oh, it is too.
It's disgusting. Look at that.
Oh, there you go. You're gonna get it. You're gonna see it. You're sick, Andy.
Look at that.
There's a biology lesson for the kids.
So the Spanish dancer is excreting, and then the shrimp is feeding. Circle of life.
Well, let's say something. I see it in the docking camera about the other. Yeah. Should cook it all stuff, so it should be able to step back. There's no kids in that.
Who would have thought you'd be watching the butt of a Spanish dancer today? I think that shrimp is waiting as patient as we are.
Yeah, that's the thing.
We're just getting footage for Dino, that's all.
That's cool.
Yeah.
You just have to keep adjusting. I'm stable. He's moving.
Yeah, no, absolutely. All right, I think it's about time. Thank you, Spanish dancer. Squishy. Squishy the Spanish dancer. That was fun. All right, so we have time for one more collection, so let's head to the shallows as fast as we can. You taking it to 50 meters? No. Okay.
I don't think there is 50 meters.
Oh, okay. All right then.
Hey, Jared, do you want to pay out something?
We're trying to stretch it out actually right now.
Yeah, it just pulled back in really hard.
That's weird. We got a belly dive here.
We could
Weird.
end the dive then.
You don't want to.
It might have been next
I don't really want to. I feel like at this stage I'm already confused what came when.
Okay.
I didn't get tugged here, so.
Yeah, we didn't get tugged at all.
Okay, yeah, it was probably as well. Sorry, guys.
All right, we're gonna get in the position. Where is that stuff off bottom?
We'll get prepped out here.
What's that? And
Okay, guys. That's all for today. We have collected, we have seen some wild things. Thank you guys all for tuning in and watching along for these past almost 8 hours. Yeah, thank you guys so much, and we'll see you again soon. Signing out from ROV Falkor.